Are we live now, Tony?
Looks like it.
Yep. Awesome. Well, we'll just consider it live. Good morning, everybody, or good afternoon, or good evening, wherever you may be in the world. Expect to see some more folks popping on here as we move through our program. This is Magnitude Live. We got a dozen. This is number 12. Since we all kind of sheltering in place doing the SIP. And our goal here is to share exciting stuff with you. It's happening beyond Earth on the International Space Station. Specifically, the focus bit has been around our ExoLab, which is an experiment that connects classrooms around the world to the International Space Station to run an experiment. And we've invited all of you, if you're just joining in, you still have time, to help us on this experiment. If we look at the scope from where we are, where we've been here since April to where we're going to be in April of 2021, is following along a single experiment, ExoLab number 8. And in this one, it's part of a series. We're going to be growing a plant up there with the bacteria, which we'll get into a little bit more. And if you guys have been to our shows, you probably have a good sense of what that is. But we're going to kind of do a review and go into greater detail about what you can do and what our program will look like over the course of a year. So we basically have evolved it into two really simple ways to go about it. You might have something that you need right here at the end of the school year. Your students are antsy. It's been difficult to do the distance learning thing. Maybe 1 out of 5 kids or 1 out of 3 kids is showing up to the class. Maybe you're getting more, which is fantastic. How do you engage your students? And there's some things we'll talk about today, like the inequities that are existing with the shelter-in-place orders and students that don't have internet access. They don't have a computer, perhaps. And there's issues with food, hunger. So just meeting basic needs that many public schools in the United States take care of. And if you're coming in from different parts of the world, I'd love to hear how you're handling that. At the back end of our program, we'll be looking at that broader question in more detail and hopefully having a pretty nice conversation about that. But what we're doing with ExoLab, and just in time here, Mr. Wilkinson's joining us. Hey, Michael.
Hey, good morning.
Now I guess you've got a class with one of your fourth grade students here in a half hour.
Yeah.
And you'll come in and join us again. So Michael's going to be popping in and out. Michael is our lead educator, director of education here at Magnitude. I'm joined right now. You'll see more folks popping in with Tony So, co-founder, and Diana Phillips is one of our co-principal investigators, hailing from Canada. So thank you for joining us this morning. So really what we're doing here is looking for a simple way to engage your students here at the back end of the year. Let's try to find a nice lightweight way to engage them, stoke their curiosity, get them excited about the future. They might be rising freshmen into high school. They might be in middle school. They might even be in elementary school and still in a sense of awe and wonder about the world. And I think it's a great opportunity for them to join us. And then the other one, besides a simple thing, hey, let's try to do something here at the back end of the year, celebrate graduation, and help us select a plan to go to space. But you might get really excited about doing the entire project, and we welcome you to join us on that. As I said, it's about a year long. We started back here in April with the selecting of a plant, and we're still in that process. So jump aboard, help us determine which legume we'd like to have as a candidate to fly up to the International Space Station. But then we're going to be going through a series of trials, ground trials and tests to figure out optimally how to grow this plant with with a symbiotic a partner, a rhizobia, a bacteria. We'll talk a little bit more today about what they're doing. In fact, Mike will probably get into that in just a couple minutes. But you can join us when school starts up again and find a way to actually ride along right up to the launch on a rocket. Northrop Grumman 15, launching out of Wallops in February is the current calendar. I think it's February 1st, but that's probably going to move a little bit because it's few months away. And takes usually a day or two to get synchronized in orbit with the International Space Station. We see the Canadarm grabbing that and going through some kind of docking procedure, berthing and docking. Unloaded our laboratory experiment, loaded up, running for about a month or so on its own, an autonomous little lab. We'll get into a little more detail what the lab does. And then it's sent home. Now, at this point, we're into March of 2021, where then once it's on the ground, we get it to the laboratory. We want probably university laboratory or professional laboratory to do some RNA sequencing to see any changes that might have occurred in a microgravity environment to the plant and the bacteria in this symbiotic relationship versus a control that we have here on Earth. In fact, technically, we have hundreds of controls because your classroom is running ground experiments connected to the space station, comparing and contrasting the changes. So for now, the Leguminaut Challenge is designed to help us just select that candidate over the next couple of weeks. And our deadline for that is June 12th, I think. We keep on moving that date back, giving folks a little bit more time.
That's it, June 12th.
Fantastic. Thank you, Michael. And why don't we dive in a little bit more about the mechanics of why we're selecting this particular plant. And I guess let me just say hello to some more of our investigators that have jumped on. It looks like Javier from Houston, working a lot with Central and South America. So thank you so much for joining us again. Shivar from South Africa has popped in. We've got Mitch and Samantha from the Lodi Unified School District in Central Valley. And of course, Dr. Richard Barker, making sure we stay true with the science. Our astrobotanist
Keeps us on our toes.
That's right. Our astrobotanist of record from the Gilroy lab. And really, if you want to go anywhere and learn the foundations of astrobotany, where they've gone, where they're going, what's exciting them, best stop on your journey, first stop for sure, is with the Gilroy lab and what Dr. Barker's doing. He's sharing with us anywhere.
Wolverines.
Oh, what's that now?
Go Wolverines.
Go Wolverines. Exactly. So, Michael, why don't you share with us at a high level? First of all, let me just do a quick review since we got more folks jumping on.
Okay.
Our sessions today are focused more back on the ExoLab and our 1-year campaign to get ready to get to space, our operations in orbit, and then what we're doing when we're coming back home again, creating more of a complete picture. We're also today going to be talking about our professional development programs are going to be available over the summer. But we'll talk here about this relationship between legumes and bacteria, why we think that's important. We'll talk about professional development. And then we're going to have a guest legume, if you will. Michael Wilkins will be talking about just one of the 9.
Right, trifolium pretacientation.
Have to get that in there, the presentation with the pretent. Yes, the red clover, and we'll use that as kind of a host as we walk through the program, so you get an idea what you might do with your own selection for a legume. But we need to figure out which one we're going to.
Not that you're going to do better than clover, but yeah.
Oh, now, don't stack the deck too heavy. We're also going to get some guests popping on from a neat little restaurant in Orlando. On your way to Kennedy, you might drop by and enjoy one of the meals from Leguminaut. They've been giving.
Yeah, if you go catch the dragon, the Crew Dragon launch later in the month, you gotta stop.
Oh my goodness, you're not going down for that, are you, Michael?
No, I wish, but I got it blocked out of my calendar. I'll be watching online.
Yep. In fact, aren't we going to do like a live kind of get together for that?
Yeah, we should. We should do a hang.
Everyone wants to do that. Just look for that Magnitude Live Plus something. Go back to space with our own stuff. Pretty cool.
Yeah. How awesome is that?
And then I'm going to do something a little interesting with everyone because I think it's been kind of like the elephant in the room. We're kind of saying, hey, yeah, the pandemic, so we're doing this thing with selecting a legume. But we're getting up toward thinking about what fall's all about and how our programs are going to work when school starts up again. I'm speaking in the Northern Hemisphere, so Shivar, my apologies. So as spring comes in South Africa, we start thinking about what's happening. What does the program look like? What strategies can we use to optimize the experience for our students that might be not in a classroom? I mean, how are you going to get all those students in your regular classroom if you've got 6 feet distance you need to keep? So what's your schedule going to look like? I don't know if anyone has an answer, but I think it'd be great to have a little dialogue on that this afternoon or this morning, depending on where you are, time zone wise. And then we're going to do our balloon launch. And Samantha and Mitch, I know from the Lodi Unified School District, are along for that. I'm not sure, Mitch or Sam, if your students are joining us via YouTube. I think there are a couple teachers that'll be joining us here in session with their students joining us on YouTube Live. They may or may not be communicating on YouTube, depending on their age and the privacy settings that they've established. Often they're going to be communicating on a back channel with their teachers through like Classroom or Teams or some other protocol that preserves their privacy, but still gets them involved in the conversation. So if some questions are popping out of nowhere, that's probably where they're coming from. So Michael here.
Ted, I'd also interject here. Teachers, if you go back, if you haven't seen some of the earlier episodes, there are lots of gems that in these broadcasts that are perfect to share with your students. They can create the foundation of some amazing discussion. They're incredibly informative. We've had some simple experiments that you could run that have been introduced. Richard's given us some amazing science. So really all ages, there's something for you there. And they're fun. So go back and take a look and see what you can find. There's some good gems in there. I know I've shared several with my students.
And that's quite a bit of a treasure hunt here. I think more than 50 hours we've accumulated now on video, covering anything from exogeology to my goodness, getting down into the actual anatomy here of the roots and their interactions with.
That's enough airtime to go into syndication, isn't it?
Maybe. We're calling this kind of playfully season 1. I think we might pick this up again in the fall with some changes based on what you guys think and some feedback. But one thing that we're really grateful for, Shivar from South Africa, and was very meticulous, and he cataloged timestamps of key points in all of these sessions that we've had. Again, this is Magnitude Live number 12, so a lot of content to look through. So we're going to be curating that in a little better way as we get into the summer with professional development. And you'll be able to dig right into plant anatomy if you need to, or exogeology, or whatever the topic may be, life here on Earth or beyond. Michael, why don't you guide us through more specifically what we're doing with legumes and bacteria and why, and maybe even talk a little bit about flight 6 and 7, the 2 flights leading up to this one, and kind of in a series.
Sure. This has been for me, and I know a lot of other people along for the ride here the last couple of years. This is an amazing piece of science that we're engaged in. Legumes are crazy important all throughout the world, every culture. We've got evidence of legumes in ancient Greece and ancient Asia and Africa. So we know that humans have been domesticating these plants for a very long time. You may know that George Washington Carver worked with legumes because in the South, the fields had been so depleted by overcropping cotton. And it was kind of a catch-22. It was the only cash crop that was worth anything, but every year you had less and less productivity. So he started experimenting with intercropping legumes and increasing the yields then on cotton years. And you've got a cash crop with peanuts. So all kinds of great things there. Some really cool history in the ethnobotany of this. And we've literally carried these things all around the world. Clover, it did not come from the Western world. It came over with Europeans as they were exploring Central and South America, and it made its way up through the migration of peoples into North America. And now we've got alfalfa as this just huge forage crop. that's really important. It's a high nutrition hay, probably one of the best hays that you can grow. But the really special thing about legumes is their ability to do something that we all depend on. Every single living organism on this planet is made up of proteins, which are made up of nitrogen compounds, which have to come from this relationship, a symbiotic relationship between legumes and a soil bacteria. We're swimming literally through a sea of nitrogen all of our lives, but it's N2. There's two molecules or two atoms of nitrogen that are bonded together with a ridiculously strong bond. Almost nothing can break it apart, except maybe a lightning strike, or these amazing little microorganisms that are able to break it apart and synthesize those nitrogen into a variety of compounds, ammonia and nitrates and nitrites that plants need. They bring that into the food web, passes it through so that all living organisms are able to build the proteins that make themselves. And in exchange for that incredibly important nutrient, literally the limiting growth nutrient of life, the plants provide the bacteria with the carbon compounds that they can't synthesize on their own. Sugars, glucose. And that's what feeds them, gives them the energy to do the work that they do. And there's this unbelievable communication that happens in the soil. If you tune into last week's episode, Richard Barker did an amazing job of going through this whole signaling and the nod factors and a real communication that happens between these organisms. And we don't think about, bacteria and plants communicating with each other, but they totally are. They're literally talking to each other and saying, hey, come here, I got something you want. Hey, I got something you want. And they make an even trade. And everybody benefits from that. So we're looking at, I just got breakfast delivered because I got back from playing tennis. And now I just lost my train of thought.
Breakfast is here.
Communications with the soil and how awesome that is.
Communication with bacteria.
So we've got this wide variety of legumes that are able to fix these nutrients when they pass it on through their leaves and fruits and flowers that we can eat, and the seeds that we most often eat as the product of the legumes. But also when the plant material decomposes, then those nutrients are getting returned back to the soil, so they enrich the soil by just growing. It's grains and fruits aren't able to do that. They deplete the soil of its vital nitrogen. But legumes are able to actually enrich the soil simply by growing in a plot of soil. So on Earth, incredibly important. If we can reestablish legumes in once arable land that's now been gone through desertification, like the Sahel or the Gobi, the margin of Central Asia, even parts of North America that have been overcropped and the soil's been depleted, that if we change and focus on more of these legume crops, we can probably bring back some at best marginal land into productive land again. And with almost 8 billion people on this rock, we're going to need a little higher level of productivity than what we can sustain right now. And if we're going to go deep into space, we've got to be able to feed ourselves. Humans are calorie pigs. Our brain uses like almost half of every calorie you consume. And our big brains are what helped us migrate and evolve, and the discovery of meat proteins. Meat's meat. Wherever you follow your game, you've got meat, and that's part of how our big brain developed. But as a result, we have this huge caloric demand, a very high nutrient demand. So we have to take that with us. And I don't know anybody who wants to eat protein bars for 3-4 years on an expedition to Mars. I love food, and I want to be able to enjoy my food when I get to the moon or to Mars and do some teaching from there, do a little bit of research from there. With these plants, we have the potential of actually converting regolith into a viable soil. At the very least, it can help us sustain our hydroponic systems and allow us to produce the legumes that give us a lot of the food that we need, but also to produce the other nitrogen intensive crops. Again, the grains, rice, corn, wheat, rye, sorghum, tomatoes, peppers, all of those things need a lot of nitrogen. And using the legumes, we can do that. I'm actually experimenting with, I love beets. So I'm growing a hydroponic beet here, but I've also seeded the rest of this with clover. So I'm hoping I can really kick up my beet and have a nice big fat one.
Pick up the beets, okay? I like that.
Yeah. So, and then I'm going to have you all falling in love with my little friend here, Trifolium pratense, red clover, a little bit later today. I'm going to go through I'm actually going into a science class in 9 minutes with my own students. And we're going to look at the actual nodules that are on these roots that the clover have produced in symbiotic relationship with those bacteria. Mm-hmm. So aren't we all excited? Everybody wants to grow legumes. I've got a whole legume farm here in my home office lab. Mm-hmm. I've got everything except I have had terrible luck with glycine max and lentils. Mm-hmm. I cannot get my soybeans or my lentils to grow. I'm having terrible luck germinating. But I've got mung beans, I've got adzuki, I've got alfalfa, I've got chickpeas, the zombies. I thought they were dead, and they totally came back to life, and they're doing beautifully. I've got cowpeas, our original leguminaut from our previous flight that we selected because they're drought tolerant and heat tolerant, and we've got really high ambient temperatures on board the space station. But the plant is a little bit on the big side, and we are limited in the cube lab to a little less than 2 liters of volume. The lab itself is 10 by 10 by 20 centimeters, so it is a 2 liter volume, but there's avionics and sensors and cameras that are in there, and the lights, so that takes up part of our volume, probably about 1/3 of our volume. So we're left with about a little over 1 liter of volume for our experiment. And then prior to that, we flew just the bacteria itself to see if it could even function in microgravity, and it did reasonably well. So the microgravity environment is not impeding its growth. It does seem to slow down its motility. It doesn't seem to spread as far, but that's really preliminary. It's a one and done experiment, so don't put too much weight in that. But that's some of the observations we made on that flight. So that was flight 6 and 7, and now in February we're going to launch flight 8 with one of these legumes, and you're going to get to help us decide that. By mid-June, we have to tell NASA this is the legume we're flying, and these are the reasons we're flying. This is how it's going to help us meet our science objectives of getting nodulation in microgravity. So I look forward to your input and your engagement in this project. It is incredible research. You will be part of our co-investigators. There is definitely real legitimate research going on here. This has never been achieved in microgravity. We know the beans can grow. We know the bacteria can grow, but we've never been able to make nodulation happen in microgravity. So this is super exciting, and there's definitely some scholarly research papers that are going to come out of this. And you and your students, if you join us, will be part of that. And I know my 4th graders are just like, they can't believe I'm going to be a published author. I'm only 9 years old. I'm going to be a published author? Yeah, because in science, we give credit to everybody who contributed to this work. It's not a one-person job. I might be the first name on the list of hundreds, hopefully, of co-investigators there, but everybody's input is just as important. I'm just trying to put the pieces together. So I'm gonna have to go talk to my young scientists now, but I will be back in about an hour, and we'll dive into some red clover goodness.
Awesome, thank you, Michael. So pull back a little bit here and make sure we've got the context right. Need something simple to do here at the end of the school year. You've already kind of feeling a little burdened with this distance learning thing and the shelter in place. Grow a plant at home is pretty a straightforward directive. And through the growing of that plant, do some observations. How long does it take to develop and grow in the environment you put it in? Preferably, it's one of our legume candidates. We've got 9 different ones if you visit our Magnitude.io website. And you can start there. If you don't have those available to you, any plant will do, honestly. If you just want to kind of think about growth of living things and a little more care about the world around us, chance to tune in and see what's happening. And the plants are always doing stuff. We're just moving so fast that it looks like they're just there again. You walk out your front door, there's that bush again, but it's changing throughout the day. It's adjusting to where the sun is in the sky. It's got its roots down in the soil. It's working and getting the water up there, and all your chemistry is happening. It's very much alive. And when you look at a time lapse of a plant, they're extraordinary. They're really engaged with their environment, just like we are. We're moving so fast in our daily lives, we miss that. So if you want to join us in the last few weeks of school, a little celebration for your graduating students, something to keep your young students interested. This is really good anywhere from K through 12. Even we've got some university students that have joined us because of the back end research potential in this flight. So right now we just need help determining which legume to send. And that is a number of simple lessons that we have on our website. Those lessons are all free. You just need to register, add your class, add your students, and walk through these simple steps, and then submit your findings. Younger students might just be doing a work of art or writing a few paragraphs about their legume choice. Our older students, we're expecting to do viable research, going in and looking at papers from others and being able to cite that work, thinking about things like the carbohydrates, the proteins, the amino acids, the minerals. What makes this particular legume better than the others, in the opinion of you as a student? So bring that to the table. That's all going to be due by June 12th. As Michael said, we're going to need to submit that to NASA. And that's kind of the end of the Leguminaut Challenge. Think about that as the first phase of ExoLab 8, a 1-year program. No cost for you to join us to help determine which legume you think we should send. Once we've determined that, here's where the fun begins. Now, school's already let out. If our students are still engaged and want to be involved, by all means, we want to give them agency. We want them to step up and say, go ahead and follow through your research this summer. We'll be active in doing research. For our teachers, we're offering professional development, and for you to move right along with us to get an ExoLab, which I want to cover here. Now, the ExoLab is really kind of the star of the show. It's enabling us to connect your classrooms, or in this case, it might be your kitchen table, with the ExoLab if your kids are still distance learning here in the fall. The professional development is right now planned around the week of July 5th, 5 days, 90 minutes a day, live video with some assignments. Pretty straightforward. It's not going to eat up your entire week for sure, but I think in terms of interest, it can probably consume your entire week with those 90-minute video sessions, and probably an equivalent amount of time on your own, planting your seed and doing your own investigations. We'll, during that 1 week, be focused on our flight candidate. So this is the legume that we have selected. Here are the reasons why we've selected it, and this is what we're doing. But there are a lot of different behaviors we're going to want to look at that plant. One of our concerns is we want to see a true nodulation occur in the environment of microgravity. Don't want to start it here on Earth. We'll have a control here on Earth in which we'll watch that develop. But having it happen in space is really what we're trying to do. So what is the right maturity of the plant to get in that rocket and set it up? We don't want it too mature because it would have already started developing its nodules, that bacteria interaction. And we don't want it too young where maybe nothing happens in the 30 days. And so what is that right time? During our professional development, we'll be introducing you to the entire concept, getting you ready for your students in the fall, and we'll be working through actual research. And if your students want to join us during that period, they can, they're welcome to. but really taking that one candidate and taking it through all of its paces. In addition to that, and this is kind of a fun extension, what is the right medium that this plant should be growing in? We've got some of our ideas, and we'll be talking about those. We've got a lot of our principal investigators here with us on Zoom, so they can share some of their methods today. In fact, I welcome them to jump in. But let's pull back a little bit. What is the ExoLab, right? And how are we able to do this? We've been very, very fortunate, and we're grateful to the U.S. National Laboratory, funded by an act of Congress through NASA, to make life better here on Earth. And the National Laboratory on the space station is it's designed specifically for the interest of the United States of America. But the International Space Station is a group effort from the planet. 18 countries from around the world helped the facilities, construction of the actual laboratory itself, the modules that are all connected to it. And more than 120 countries have run experiments. Almost every country on the planet has run experiments on the station. And really thinking about how we can make life better on Earth. What happens when you take a force that evolution has taken everything and evolved with, this gravity? What happens when you remove gravity from the equation? And it's not just living things. What happens when you remove gravity when you're dealing with magnetism or microfluidics or some other physics phenomenon, chemistry interactions? Gases behave differently, liquids behave differently. So this investigation really has us looking at the world in a much different way. Well, it's really hard to do here on Earth, but in this microgravity environment, we can investigate some very specific things. What we're looking for with plants as a whole, and Dr. Barker, please weigh in, is genomic changes. The genes, the plants go under stress, or a living organism, humans, astronauts go under stress and their bodies change. Our plants change too. And when they change, they trigger different proteins. We can learn a little bit more about these plants when we're put in those conditions. I don't know, Dr. Barker, if you want to just say something a little more eloquent than me about that.
I could show you a little bit of data from a space study, and then talk a little bit about time-lapse photography and how it's fun and easy and a really important tool for the students and the teachers. Does that sound like fun?
It sounds great.
Yes.
And then we'll walk through the ExoLab itself.
Yeah, I think this should lead into that hopefully quite well. Ooh. So where were we? Kara. Yeah, I was just having to be looking at some spaceflight data. This was done by Rob and Annalisa Paul from Space Plants, Florida. And they've been looking at little plants, Arabidopsis plants, and they got to sellotape some of them to the wall of the space station. And some of them they put in like bags, so they were in darkness, and some that were in light. The cool thing is when they harvested them, they got the astronauts to take photos. And so they've got a couple of different varieties, a variety from Poland, a variety from America, and a mutant that can't see some types of light, 5D, phytochrome D. I think that's red light. Okay, so it's been a while. So they can't see a particular frequency of light. But the astronaut was taking these pictures, these photos from with a camera, a digital SLR. So taking them at these strange angles. Can you see how that's not like straight on? Fortunately, with Photoshop, you can kind of realign things, but it takes a lot of time to do that. So, so if you've got your camera like in the ExoLab where it's perfectly next to your sample there, they're aligned to each other, it makes the analysis of the images easier afterwards. So if some of your teachers are going to try and convince us that their type of legume has the growth patterns that would be most interesting to observe, a way to kind of show your peers, a way to show you the other teachers and the rest of the Leguminaut team would be time-lapse photography, because this shape, this structure is going to change over time, and it's going to change depending on the environment that you put it in. And so I like to use time-lapse photography because it kind of shows me the environment. It kind of shows you broadly like through time, and then you can choose the sweet spot. And that's always like the key, like when you dissect the plant to look at the nodules, when you take, and because if you look at it, because generally every time you dissect the plant, you're damaging it, right? And so time-lapse photography is a non-invasive way to get these information. And if you've got really detailed photography, really high resolution, you can start to see changes. So even just looking at the width of the root of these plants in orbit, we were able to see one of the varieties has a slightly wider root in orbit. It's slightly fatter. And that's probably a sign of stress. So the moral of the story there is photography of plants in space. By looking at them really closely, we can see new awesome patterns that we just don't see on Earth. And so how do you get into a situation where we can do this regularly? This is the journey to becoming a astrobotanist. Yeah. So we talk a lot about the journey to Mars. And I once got asked to talk about how I got involved in this journey. And you know how I like to go back in time. Well, I've added this slide, thanks to Javier, because my passion for time-lapse photography that brought me to this science and that brought me to this awesome journey that humans are going on to Mars actually started with a game called Capoeira. So I was playing this game, I was doing this martial art, and I wanted to learn more about what was happening in the game. When you're living in the moment, time goes so fast. And so I realized by filming stuff back, I could watch and learn and see the mistakes that I made and see things from a different perspective. So perspective is everything. And now we're thinking of things from the plant's perspective. So they're moving in their own time zone, a bit slower. And so I needed to start to change the photography in order for me to start to appreciate how the plants were going so I can understand more about them. So the skill I was developing in my hobby suddenly helped me in my work environment. So I started to think about changing the exposure time of my photography. And this then helped me with another hobby that I was developing at college, which was spinning fire. So if you increase the exposure time and you have the right frame rate of when you take your photos, you can start to see different phenomena. So the video of this itself, you just see these tiny dots moving around. But by using time-lapse photography and by changing the parameters, you can actually create different patterns. This is a great thing to do with students. You might have seen like light graffiti, where you get the laser pointers and you run around. It's a really fun thing to do. If you use the green laser pointers, you can point all the way up at the moon and the stars, and still also use it to practice light graffiti and less time-lapse photography. So making it fun, turning it into a game. But practicing that over and over, eventually you get to a point where you can start to extract data from these images. And this is an example from a paper that we published in Plant Methods a couple of years ago. Here you can see we have a happy plant. Every single day we took a photo and you can see it getting bigger over time. And being a cruel person, I decided to torture the poor thing. It's one batch I didn't water. This line here represents the day when the stress began. And another batch I added salt because I think salt is really important, which we've discussed in previous things. And what we can see is over time, the shapes of the plants change. And you can see that with your own eyes, but as a scientist, we have to quantify things. And so, but because we'd practiced the time-lapse photography before, we had a stable system. And so we were able to measure the size change. And there's different ways you can measure the area, the diameter, the perimeter. Usually where I start with students is just having them draw a straight line across and just doing that a bunch of times. Like that's what I call my ground truth data set. You have to look at each plant with a ruler in your hand. It's great. But eventually you can do that just with photos. And then because we got excited, we managed to meet some NASA researchers, and because NASA a bit like working people who get really excited about data, and we were, so we found these collaborators, and they have what they call a hyperspectral imager. So we normally look at things with color. When we take photos, color is in the camera is usually an average of RGB, red, green, and blue. And so your cameras tend to have each pixel tends to be a combination of those three values. But with a hyperspectral imager, you can essentially make each pixel have a numerical value for each frequency of light. So you can create thousands of different measurements for each pixel, measuring all the different frequencies of light. And this is what NASA do when they go to distant planets and stars. This is spectrometry. This technology was developed to look at distant planets and stars. But now what we're saying is we can turn that technology around, look back at the plants in the room next to you, and then you can see things that we can't see with our eyes, but insects can, because insects see in ultraviolet and infrared and these other parts of the spectrum. So by collaborating with NASA, we got to use this really advanced imaging device to be able to look outside of the visible spectrum. And then we had, in order to do that, when they use this device, they usually fly on airplanes and spaceships to kind of look down on big surfaces. So to use it in the laboratory, we had to build a robot to move it back and forth. And so those designs on the software and the hardware are all on GitHub, because that's how scientists share the protocols they develop. And now researchers at the Kennedy Space Center are trying to develop this technology there to help them develop strategies for on-orbit cultivation of crops. But not everybody has access to $1 million spectrometers that NASA fly around on airplanes. And this was a rare opportunity based on me finding one of these scientists who only used it half the year. Half the year it was on the airplane, and half the year it was in their cupboard not being used. So we essentially used student ingenuity to create a new device on something that was not being used in its downtime that was more valuable than my house. So just trying to make the world more efficient. And so those designs are there. But it's not very accessible. And that's one of the great things I found about the ExoLab. When I plugged that in and turned that on, it was really quick and easy to get operational. It's really accessible. And that is awesome because if more people can access these technologies, then that means more people can understand the data that's coming from them. And one of the things ExoLab does is similar to what this WiseCam does, which is time-lapse photography. And this is my favorite way of observing the world. So this is a tool, this WiseCam thing is produced, it's like $20. The cam, you have to add a memory card to it. The early versions, I got the first ones like last year, and like there's glitchy in the software, and there was an article about them losing everybody's like security codes and such. But essentially it's a security camera that's been made for people's houses. But I've turned it around and made it and pointed it at my plants and seen that it does actually work. So this is like a fun, cheap way to practice time-lapse photography and also educate students about like people being watching you. You got to be cautious of that kind of thing. So I'm in the process of, again, working with some academics to try to develop software to extract numbers from this to measure plant growth. But you can see there's a slight curvature there based on the fisheye that comes with this camera. Oh yeah, this is the thing. So I don't have stakes in the company or anything. It's just a really easy tool to use. It's really cheap. And the real value is going to come in communities working together to use it and working together to work out how to process the data in an efficient way. I think that's the real value there. And teachers are the perfect people to experiment with that. And a educational community like Magnitude is the perfect way to coordinate such groups, in my opinion. And so this is a figure from a paper we've just published using a very similar technique where we had this kind of fisheye effect from a Raspberry Pi. And my friend, the manuscript's just been submitted. And I mentioned MATLAB has a code which can deconvolute the curvature. And this was them demonstrating that, practicing that with a checkerboard in the middle. So work in progress there. And so why am I raving on about this whole time-lapse thing? Well, you know, my roots comes from like trying to understand plants, their growth, to help the future. Okay, this is my favorite one. This is, okay, so what they were talking about then was like loads of cameras. Everyone should be practicing time-lapse photography all the time with their smartphone. People should be working with teams like Magnitude to coordinate like large time-lapse experiments together so we can find big patterns. But when you don't have a big group of friends to collaborate with, help, sometimes you're left by yourself, a bit lonely. And this was me at the end of my PhD when I had lots of plants I'd modify and I had to understand them all, and I didn't know which time point to look at. So I got really interested in time-lapse photography. I'm really interested in robotics. And so we built this robot. This guy's called Osimo. He's named after Cartman's robot from South Park. So it's going to load, it's just slow. So basically we took one digital SLR and we moved it around a lot. So this is it working away in the lab, night and day. And we're able to photograph for lots of different plants. So I thought the robot would liberate me. No, I became a slave to the robot. I really did. To keep this thing running, to process the images, all this stuff. which is why I'm a huge fan of the concepts where you have lots of teachers, lots of people working together to essentially each have their own camera, so to speak, and do their own like sort of metadata analysis, and it's better that way. But we did get it to work, and again, the codes and such are online on my GitHub account, and we were able to measure the roots and the shoots. The roots went down to, we grew some plants, we put them into the machine on their side, what we call a gravitropic assay, and the roots went down towards gravity and the shoots went up towards light. And so that was great because my old supervisor, Patrice Masson, was able to use these data to justify putting Brachypodium station onto the space station. So this was the astronaut Scott Tingle, the week before he loaded the Brachypodium samples into the veggie. And these data are still like work in progress. Like he'll be publishing that in the near future. So this is just the opportunity. I often rave on about my hero, Arabidopsis thaliana, the true Groot of the here and now. But Brachypodium distachyon is his cousin, which is a grass, some monocot. So it's closely related to rice and to wheat, and the fundamental basis of the food system that feeds like cities. So we can by understanding these two like cousins, and we can hopefully understand all plants in a better way. They have the same number of genes, the same number of chromosomes. These two species are really similar to one another, which makes them great model organisms. So when you have a good model and you have high throughput system, if you get the experimental design right, eventually you see something unbelievable. So this guy here, yeah, so gravitropism assay, lots of robotic, so high throughput, roots all bending down towards gravity until they don't. So down is the gravity vector. But look at this plant, isn't that awesome? So this was from my friend Edgar Spalding. His lab is the one next door. They were the first lab to ever do robotic photography like this on a large scale. And a couple of years ago, after like extensive mutant screens, they found some varieties, some mutants of Arabidopsis that have roots that grow away from the direction of gravity. Mind blown. How is that possible? I don't know.
They came in via meteorite or something, right?
Okay, so time-lapse photography is awesome. It's great. It's the here and now. But we want to go beyond state of the art because we like to work with like NASA and such, right? And so they've always been thinking futuristic. So I've shown you previously this fluorescence photography as well, so this is still just time-lapse photography. It's the same thing. Now we're just illuminating it with one color of light, blue light. And we have chemicals in the plants that then glow green when they get illuminated with blue light. And so we can see inside the plant, and you can see that fluorescence, that glow, that's the plant's nervous system. So this is just a very hungry caterpillar getting its dinner, and the plant screaming and activating its defense system. And this was made possible because, so think of the plant there, you're shining a blue light down on it from above, and then you're photographing it down on it from above, from behind the blue camera. Now imagine you break that light into a laser beam, and you put that laser beam through a prism. So now you have like a sheet of light, and you're moving that sheet of light rapidly across the surface of the organism, and you're moving your focal plane with it. So essentially what we have on the left here, this is what's called light sheet microscopy. This won Nature Methods of the Year for the last like 3 years running. And okay, this particular video isn't the most impressive video in the world, I know, but I did it myself with the help of a collaborator, Henry, his group of people that invented the technology. But so this video here is actually 300 gigabytes in size. Okay, so we collapsed it down to an AVI so it can be watched. But why is it so big? That is a ridiculous size for any data set. Well, that's because they can make a three-dimensional model out of it. Because they've been shining this laser sheet across it, they've been scanning it. And because Arabidopsis roots are small and thin and don't have too many like chemicals in them, they can actually build a three-dimensional model of it. It's just you have to have supercomputers to process it.
Almost like an MRI in a way, isn't it?
Oh, sorry?
Kind of like an MRI.
Yeah.
Conceptually.
Exactly that. The interesting thing, the difference would be, or the main difference would be, by changing the frequency of light, you can excite different chemicals that are in there. The MRI has works exactly the same way. You're totally right, and it just but that's worked on a very specific frequency. But with the light sheet microscopy, by changing the frequency of light, you can hit different chemicals or different fluorescent proteins or whatever. But here's the really cool thing: if you look really closely there, maybe I've just looked at it too many times. Can you see that? So this is just a plant growing, and I think this is its nervous system, because I'm crazy, as you've probably gathered right now. And it's just kind of just slowly growing. So we haven't provided any all the other times I look at the nervous system, like the response to being eaten by caterpillars and stuff. That's a really extreme stimuli, and this is just the plant growing. And what we're seeing here, can you see this little flickering, these little pulses and such moving through it? Well, this is that electrical signal. So we've got that electrical sensor in this plant. And so basically, as plants are growing, their surfaces of their membranes are constantly tingling with electricity. And when you give them stimuli, ding, it sends signals to different places. And that's kind of why I'm a huge fan of time-lapse photography, because as you can see, as I've gone through my different stages, well, I think that is, yeah. And as you can see, as I've gone through the different stages of my life, because this was for that high school.
I like how Fire Dancer is just in there. Boom.
Right? So when I was young in high school, I just wanted to be an environmentalist. I loved growing plants, and I liked juggling fire at raves and festivals. And as I move on, I realized that I wanted to continue to focus on the skills I was developing, and yet let them mature as I was supposed to. So my passion for environmentalism became a passion for agronomy. My passion for horticulture became a passion for just plant science in general. My love of fire dancing became my passion for time-lapse photography, because I wanted to observe it and reflect on it and find ways to improve that iterative self-improval, that grit, you put into everyday life. Agronomy evolved into molecular biology and cell biology. If you want to understand the size of your yield, you need to understand how the cells move nutrients around. The plant science and everything became genetics and genomics. And my photography became microscopy, just focusing ever closer in with ever more complicated tools. But essentially, it's still the fundamental basis of time-lapse photography was the skills that allowed me to enjoy microscopy. Molecular biology unfortunately evolved into statistics, and I think I did really well avoiding it that long. I think most people that go into biology because they heard it's the science that has the least math. I was definitely part of that category, but eventually the statistics became part of the way I work, and that's thanks to modern advances in computer science, which is why I'm so jealous of this generation who get introduced to Python early on and the Jupyter notebooks. Because it's no different from writing it in English or French or German. So it's just being introduced at that early stage. Oldies like us, we can learn those languages. It just takes us a bit longer. So which is one of the reasons I love to collaborate with students, because they help bring me up to speed, and they really do. And so finally, you can just basically see how life isn't linear. It's a bit chaotic, and as you go through things, some things link onto another. But ultimately, all of these different skills and hobbies have all merged together, and I'm now given the opportunity to work with NASA, and now also collaborate with Magnitude. Because so thanks for letting me try and get people excited about time-lapse photography, and let me introduce the WiseCam as another tool in the arsenal. And it's at the price point where it's totally accessible. And the one thing the teachers would want, because there's an app on your phone, you can download the time-lapse really easily. And eventually they would come back with the images, and we would then need to work out a way to extract data from them. The video in itself, in its own right, is really valuable. Just watching it back and seeing that's a huge data point in its own right. But if there was interest, we would also be able to develop image analysis systems from them.
Well, that's fantastic.
All fun and games. So the finding before I pass it back would be, so you guys, as part of the Leguminaut challenge, you guys are now developing data, and by growing your legumes and by changing the different environmental parameters, the types of soils that you're using, these things, how much inoculant to add, all these sorts of really important things. And you're probably recording those down in a notebook, maybe starting to digitize that into a spreadsheet. But at the same time, maybe you haven't considered doing some time lapse, because when you present these data to your peers, your friends, you need to help them understand the context. And showing someone a photo of what you did, showing someone a video of what happened, says a million words. And so you can get away with a much simpler, cruder table and a much shorter write-up if you've got a good photo and an all right time lapse. And that's how I get away with it.
Well, and a little bit more too, I think. But yeah, thank you for that journey. Always sharing a little bit more. The fire dancing, that's pretty awesome. We'll have to with the capoeira mix in there. It's like
when are you doing that again? The fire dancing.
I'm available. I do weddings, funerals, bar mitzvahs, any opportunity.
Classroom investigations.
I have done it on a couple of educational programs at UW, like the summer, high school kids that came in. One of them that actually ended up setting me up with my wife, different tangent. Then actually they found out about this hobby and they spoke to their coordinator of the camp and they dragged me out and it was so much fun.
Hey, how about a high school graduation for my daughter?
I'm there, yeah, definitely. Give me the flight, I'll be there. I'm bringing my equipment. I haven't lost like riding a bike.
Oh, there you go. Well, I'm just curious what that might look like in microgravity.
Well, have you seen the video? You must have seen the combustion experience.
Exactly. Yeah, it's pretty amazing. The what's beautiful about that time-lapse photography is we are kind of slowing down to observe the plant in its sequence of time, right? It's got a different meter it's running on. I guess chemically it's all doing the same thing. It just takes a little bit longer to do what it's doing for us to see anything noticeable. But their lifespan is much shorter than ours, right? So I guess we move kind of slow too. Time lapse for me as a kid to now, that would be pretty horrendous to look at, but you'd see me aging and what have you. So I guess if you have a camera pointed long enough at something, you can discern that. And plants move pretty quickly through space-time, so it's nice to be able to see that. Exolab is one of its primary components. I guess I should show what an Exolab looks like. Is the camera. It uses a Raspberry Pi fisheye, and it captures an image once an hour, whether it's a weekend or a holiday or a pandemic. If you've got something in the classroom that's running, it's still running until you've decided to go in there and turn it off. Exolab, mine is plugged in right now. I'm just going to disconnect it right now, and I'm running a trial for upcoming. We're trying to find out which legume is going to be the right one, but we can get into that a little bit later. But the mechanics of the Exolab, and many of our teachers already here are working with it, but for those of you joining us, it's just a kind of an overview. This is about 20 centimeters tall, about 8 inches tall, and it's 4 inches by 4 square, or 10 centimeters. This volume here is designed around the same lab that goes to space. This is what happens in your classroom. And this really has been developed around a form factor called a cube lab. This is what they look like when they go to space. So this little guy's been in space before. It's just a 10 centimeter cube. And if you look at our Exolab, you can see it's essentially a 2-unit equivalent, or 2U equivalent. These labs come in a lot of different sizes, but this is pretty much the form factor. If you can put it in the box, so you can get it to space a little more economically. I guess the best analogy is that 40-foot truck you see on the highway or on the rail cars or on a ship coming into the harbor. That's intermodal transportation. And we can fill those up, send them anywhere in the world for a few $1,000s, which is pretty amazing. I wish we could do that for space. Our intermodal transportation for space is in this size. It's much smaller, and it's a lot more expensive. But what's nice is the cost has come down versus building custom experiments. So if we can fit it in these labs, they're standard docking modules. It's missing its pins right here, but for power and for data, for imaging, what have you, computer processing, it's all kind of self-contained. So that's the reason why the lab looks the way it does. Inside, we'll have our experiments running. But here's the star of the show. Dr. Barkley was just talking about a camera taking a picture once an hour. If it's in a classroom setting, you're worried about privacy, the LED lights are actually going to flash a minute before the image is taken. And we even have a cover that can go over the lab if privacy is a big issue for you. It gets that range there, it gets a little fuzzy out past what we're really trying to look at, but still, I mean, there's some issues, so we take that into account. But in addition to capturing an image once an hour, it's reading the ambient information of the environment around you. So we've got a carbon dioxide sensor here, something that's the food for the plant right there, the CO2. So let's see, what are the levels? As well, I guess I should talk a little bit more about the temperature and humidity are other sensors that are here. And this little thing sticking out is a light sensor, because you can adjust the wavelength of light, the duration, and the intensity of this light array. So if you want to illuminate your plants under a red light for a week to see what might happen, or a blue light, or a green light, or a white light, broad spectra, you could do that and then observe those changes. What's nice here is this is an Internet of Things device. So as it's taking an image, very similar to that WISE camera Dr. Barker showed us, it was really designed for security. This is designed for plant growth observation. We're taking all this information, sending it to the cloud, and then we provide a dashboard where you can see your plant growing in space, your experiment, or rather in your classroom, with the experiment that's actually growing on the space station. And so, Tony, I don't know if you want to pop up our classroom platform just so people can see what that looks like. Here's the fun part. Diana's in Canada, right? I think we've got some folks here in Northern California. Michael, when he comes back, is from New York. Shivar is here from South Africa. We've got Japan, Germany. We get schools around the world. They're all interconnecting their information. So it might be really cold in Canada and nice and toasty in Central America somewhere. And so that ambient temperature is going to affect, and humidity, and maybe the light levels you've chosen for your plant will affect how your plant grows. And we can actually see them side by side. So you can compare your plant or your experiment with what's happening in space aboard the International Space Station when we launch in February. But you can also see all of your colleagues' plants from around the world and what might be happening to them. Now, it's pretty difficult to have everyone start at the same moment in time. You've got your scope and sequence that you're introducing your students to. You might be starting weeks later or weeks earlier than somebody else. But what we do at Magnitude is we synchronize everyone to kind of a meter on the space station. So when we start our experiment in space, day one, you could start right around the same day. We usually ask about 3-day wait time, so everything is synchronized. But you can start weeks later if you wanted to. You will only see day one when you first start, even though it happened 3 weeks ago, for example. Day two, across our entire network, regardless of when someone started. So if you're the first one to get started, you don't really have much to compare to. But if you wait a couple days, then you'll see what other people are comparing it to. And if you're kind of you're getting started a little late, then you can look at everyone's plants on day one, hundreds of plants you can compare. And of course, at the end of the experiment, you can see it all, whether you're doing the time lapse, you're looking at data in a series, or what have you. So Tony, do you want to pop up the Magnitude platform?
Yeah, I'm looking for one that has images.
Okay. And what I'll do while he's doing that is just kind of show you what we're more than likely, this is the experiments on Earth are going to be growing in these size tubes for Flight 8. I think that's what's going to happen. We're still working through our flight manifest provider in NASA to confirm the volume. These are 38 by millimeters in diameter by 200. And what's going to go to space will be a little bit smaller.
I can resist.
Oh, there you go. He's moving fast. Well, our plants move slower in time, and we move at normal speed. Dr. Barker, you must be at like a different
I already had it open because
there you go.
I'm new to this, but this is awesome.
Actually, you want to bring up Gil's. I just found his. His is looking really good.
I don't know where his is, do you know? Or do you want to? You can feel free to take over. Like I was just playing.
Okay, yeah. Let me share my screen.
Yeah, go for it.
So what's interesting here is the ExoLab during the Leguminaut challenge is not required. And so you'll see a lot of those labs are not really occupied with anything. And we have a lot of teachers that don't really know much about ExoLab right now, and they're just trying to have some fun with their students. So you don't need it to get started. We need help finding that legume. But when you are underway and you're running some trials, this is what it might look like.
So what you're seeing is a experiment from Japan.
Looks good.
Okay, well, this is how he started. And this little guy is starting to come up. And you can literally see the entire history of your experiment. Wow, that grew really fast in one day.
It looks like the agar is quite hard.
You think so? Okay.
Well, I mean, it's surviving, don't get me wrong, but I reckon it would, if the agar was a bit softer, that would go in a bit nicer.
You can see it's working the way it's on the top. It's trying to push down.
Yeah. It needs to find an anchor. It's going to have to get its whole way, grab all the way around the edge to be able to get in, I think.
That's really interesting. It's exploring right now, right? It's trying to find a way in.
Mm-hmm.
This is brilliant. And this shows you the strength of this system, right? Like, and this is, you're getting to go through, which time point would have been the one to look at it? Who knows? Like, do you go look at your plants every morning, every afternoon, every evening? It's impossible because you've got so many different things you want to try. Like, and to have this like defined system so you can skim through and go, oh, look, well, it's getting down the edge there on the one on the right. You can see there's some roots, but that's squeezing down the edge there.
There's some happening over here too.
Yeah.
So you can actually click on the image. And
you probably won't see that because it's a pop-up. You might just be sharing your window.
Okay.
But having said that, having a hard agar is not necessarily a bad thing, because if it forces the roots to the edge, then you will definitely see them if the nodules are there, right? But then there's going to be that trade-off. Like if it's softer, it may be less stressful at the beginning. So it'd be really good if the teachers did a concentration gradient of agar, like some diluting it a little bit to see. And that's what I would do. But
I've been starting to dilute from the stock recipe. It also helps my product go further. But
about 50% dilution.
Yeah, about that. Not quite. It's more like about probably 30 to 40% dilution.
That's a good idea in terms of overwatching.
I just add some distilled water before I boil it. And it's just a little bit thinner and I get a little bit of root penetration.
Does it come at 2%? I think it comes at 2%, right? So if I tend to use about 1%. I did this crazy study with a wonderful student, proto doctor Natasha Bilkey. She's doing a PhD now in canosensing in plants.
Proto doctor.
I'm so proud of her. She's amazing. I should get her on here. Like anyhow, she compared 32 different media types. So she went through gradients of hardness and quantified like skewing patterns, like growth rate patterns, like length, density, that kind of stuff. I'll dig these data out and send them over, make them accessible. They're buried away somewhere.
That's wonderful. And that's a great prelude to our summer programming where we're going to be looking at materials, and we can do something like an assay to look at the different concentrations.
Exactly. So it's great you like recognize, like watching the news and stuff, like really start stressing.
The other thing that made that experiment so big was we also compared agar to phytogel, because there's different gelling like agents. And that had a minimal effect, but you are changing the nutrients components. So like,
I don't.
So one of the things that we can say is based on that study, you can rule out the use of different gelling
Find out what you need.
just select one, so it wouldn't have to be as big. I think there's data now. It's here somewhere.
Oh, there's some nice root development.
That's a view. Can you see any nodules yet?
Yeah,
I don't think those are inoculated.
Yeah, they don't look like it, do they? I know he has something that he got nodules with.
So these are Gil Suzuki's, is that it?
Yes.
Yeah.
What about the right hand one? That might be at the top.
Oh yeah, back behind. Yep. Good eyes.
So can
looks like it's in the right place, and up underneath that arch that goes up like an aortic arch If you follow it up and to the left and then part comes down, there's a spherical object right between the first and second. Yeah, just a little bit lower.
Wow.
That's what you
This
want.
is like Blade
Yeah.
Runner, right?
Looks like that might be
25,
enough.
135.
Right here.
So one of the things we often do in the laboratory, we'll do lots of experiments changing on the different variables like media, like concentration, that. But then there's the experiment that goes into the flight certified hardware. And that's what the ExoLab is. It's flight certified hardware. And so once you think you have the protocol that works, that's when you load it in.
Oh no, really?
And so Gil had went through that exact process. He had some prototypes in his home, in his lab. And then when he thought he'd got it right, he put it in there. And so now we have the time point when we saw those nodules. That's a really great like milestone, so to speak, because it's nodule formation in the flight certified hardware without contamination. So I think that's and it's useful to then provide that summary and that metadata of how that was presented to the other like people who have other candidates, so that can be compared and contrasted. In my opinion. So I'd recommend. Because yeah, that nodule, that was cool. It's so cool the way you can look at that.
So the visualization is really important. Again, with the distance learning and what have you, your students can follow along the experiment. They can dial in and look at it on the weekends. I said dial in, didn't I? My goodness, that's old. They can log in and I know, right? 9600 baud guy running this morning here. I was up too late last night. But so right now we just need your help to select a candidate to go up there, and we have till June 12th. You've got plenty of time. Just go out, and I went out and just had a bag of red beans, and I got some of them, and these little guys germinated on me, which was just a bag of, look at that. It's like I'm ready to plant these guys.
So yeah, it's been
excellent.
So it's not like a big expense here for your students to go out and do that. They probably have something in the cupboard at home. Whether it's a candidate or not, almost as irrelevant in my view. It's that they're involved and engaged. They're thinking about life. They're taking some observational measurements and what have you. We will need to select that candidate. When we're talking about this media and what the proper media to go in is what we're going to be doing this summer during a 5-day professional development from July 5th on. And this will lead right into our fall programming. And for those of you really looking at something that you want to kind of grab onto, introduce your students to real world science right at the very edge of discovery. This will be running really all year long through April or so next year as we go through these different stages. So that PD is just part of it. We'll probably open up a season 2 of something like Magnitude Live, in which we can get together once a week and talk about our findings. We have a pretty active community right now of our researchers, and we welcome you to join us in that. Let's see.
This is a really cool. We are looking at images from Japan and New York at the same time. I'm sitting here in Berkeley.
Hey folks, I don't know if you can hear this, but your voices are getting overridden by a conversation on the phone, a female voice.
Oh. It's not from here.
No, it's okay. It's the work at home thing. We've got multiple adults working. It happens, I think, and so it's part, I guess, par for the course.
That sample from New York that had the powder on the surface of the agar, was that the inoculant?
Yes, that should be.
Because that looked like it'd be a really quite a high concentration. Whenever I try and add inoculants to confined environments, I read what it says on the back of the tin, and then I do a concentration gradient down from that. Usually, like the recommended retail volumes of like beneficial microbes for like tomatoes or whatever is usually a little bit higher than you want. And if you have a too high inoculant, that can overwhelm the legume. So like symbiotic relationships, well, they're symbiotic eventually, but there is like that molecular dance, which which is the borderline between like zoonosis and pathogenesis.
Right. I think we've seen
so-
pathogenesis more often than not.
Yeah, it's a thing, right?
Yeah. Well, I guess it's about really doing some measurement now. It's like, oh, we'll sprinkle a little bit on there. That looks pretty good, right? It's like, and
Yeah.
when to do it, right? Do we roll the legume seeds around in that before we plant? We put it and mix it in the agar. You know, do we mix it in some other media? Like it looks like.
I mean, there's definitely a seal or dilution that someone's got to do, I'd reckon. So you'd get the initial inoculant, you work out the colony forming units per like gram or whatever, and then you like do a titration, I think they call it, where you dilute down again and again and again, and then you do that gradient and you just have to do the comparison. And some of them, you'll just get microbes covering it so quick, you know, nothing else would survive at that concentration. And then there's a sweet spot, and there'll probably be like 2 or 3 dilutions that will work fine. It's just finding that rough range.
There you go. And we can add that to our PD for the summer, as well as some programs during the school year. There are just so many things that we can investigate here, directly and indirectly related to the mission itself, in terms of finding that right Goldilocks zone, right? Not too hard of an agar, not too soft. Right? Too much inoculant, not enough inoculant, right? A plant that grows too fast or one that grows too slow. And so all of these conditions have got to be met if we want to get this nodulation, this dance between the rhizobia and the legume that we end up as a flight candidate. And then, you know, other considerations, light levels, right? We have kind of fixed with our carbon dioxide and temperature levels with what's going on in the station. And we're reliant really on the ambient temperatures you have wherever your exolab may be, if you're going all the way with us to the flight. But so those are variables, but you know, how do you control those the best that you can? And so as we have hundreds of classrooms working with us on a normal flight, what's nice here is we can find that range and we can see where we've gone a little bit tipped over the range, and we can see where we kind of tipped under it, and we can find that sweet spot, and we can look back at what someone has done through the time-lapse photography. One thing that Tony didn't share with us is we also have data points as we're averaging the temperature and the humidity data over time. I think we get 5 readings an hour, and they're averaged. And so you get this nice sequence. You can see the binaural cycles of carbon dioxide. If you're in a classroom, you can watch it climbing during the day and you watch it kind of going down at night. And those are observations that we don't need to make directly, but we can ask our students to investigate those and wonder why, right? Why is this cycle occurring? And for them to make their own hypothesis and then find a way to prove that. One interesting thing we've had some folks do, Michael, I don't know if that was you doing the Alka-Seltzer. Was that you that did that? Right? So they wanted to bump up the CO2. So the little dish, put a little Alka-Seltzer and then put it inside the exolab and then watch the CO2 levels go up.
Yeah, they tried a whole series of things. First, they were exhaling into, we set up a tube and they each had a mouthpiece and they kept and exhaled until they about passed out. And we could see the carbon dioxide level rise, but that wouldn't sustain. So then they were brainstorming different ways that they could get a sustained level of it. And they tried Alka-Seltzer, and that worked, but you had to keep feeding it. And then we actually experimented with a yeast culture, and that did well until the yeast just totally overgrew. We had it in a 1 liter bottle, and then it was off gassing into the lab. And then we got really sophisticated and got those little 16 gram CO2 cartridges and a regulator, and we just kept a nice steady infusion of carbon dioxide, and we were able to keep it around 5,000 parts per million pretty steadily. But as memory serves, then we were also trying to elevate the thermal environment, trying to match what the conditions were on board the station, and then we ended up cooking our display inside the exolab. So we had to replace that.
Yeah, you get a little too warm there.
Yeah.
Pretty interesting. Wow.
But it was a great process of trying to verify that was what was killing the plants were those conditions. And we did in fact see the same kinds of results in our exolab in the classroom as we were seeing on station.
Hey, you know, there's a question that came in a little earlier. James Cottrell asked it about Mars. Where'd that question go? We didn't, let's see. Legumes that produce beans are typically full light plants, meaning they need a lot of light. On Mars, the light level is about half of that on Earth orbit. What bean producing legume species can handle low light levels? Sounds like an investigation waiting to happen.
Yeah,
right?
that's a great investigation.
And there's two aspects to the mission here. You know, we're looking for that nodulation, so we get that nitrogen fixation between the rhizobia bacteria and the legume. That's our flight candidate. To perhaps create a garden on the surface of the moon or on Mars. Garden on Mars sounds pretty awesome. But really, life here on Earth, we would love to make it better. And so we feel very strongly about the United Nations Sustainable Development Goals. They've got 17 of them, and we really believe in all of them, but we're going to select a couple. You know, equity, super important. Everyone should have an equal chance to do things like have a roof over their head and food in their belly and a good education. Quality education is another one that the UN is supporting, when we do as well. And the third one is zero hunger. There shouldn't be a world in the 21st century where people don't have food to eat. And we're thinking about right now, the pandemic, well, you got to have a job. Many people don't have a job. It's getting up to 1 in 4 here in the United States, and around the world, not getting much better. So let's start thinking about what we can do to develop our own food resource. What does it take to grow food? So again, going back to something super simple and fun with your students just in the last few weeks of school, grow a bean, grow a legume, grow any kind of seed you might find around, and think about what it might take to sustain yourselves. If you haven't created a garden, perfect time to start thinking about what might be required. Diversity. We have monoculture now. There's like one kind of corn, right, that you can get, one banana. So what can you do to help kind of create diversity? And I'd love to meet this gentleman from Ethiopia, the research that they're doing in Ethiopia, that Dr. Barker introduced us to. I guess Alex his wife just had a baby, so we'll probably have to talk to him in a few weeks. But the idea here is that these little farms have diverse crop, and the farmers know. They kind of have that little culture of generations of like, use this bean this time of year and what have you, and they share and exchange. But there's a diversity of crop that exists versus like a single, hectares and hectares of the same kind of crop, in which you have disease issues, right? You've got over saturation of certain nutrients and what have you. So that diversity really helps. Just like you got 8 billion different types of people on the planet, right? We're not all clones. Which makes a beautiful world, right? And so I think we can look at that as the plants as well. So how do we make life better on Earth? That's the main driving mission of the U.S. National Laboratory. Again, which we're deeply grateful for. Without the National Laboratory, funded by an act of Congress via NASA, we wouldn't be able to reach out and do these fun experiments with you guys. So the ExoLab, not required to get started, but if you want to join us for the whole mission, we're setting up a real special program for you guys now. You can learn more about it on the site. Think about a cost of a field trip. Take the number of students you have, divide it by this. You can take a field trip to the space station this year for probably less than $5 a student, which is pretty amazing, right? So I'd say check it out and get involved. Michael, you want to kind of dive us in and taking one of our legumes. And if you go back into some of our earlier episodes, and we're really going to have to catalog those a little better, you can see these other introductions as well. But we're going to use clover as an example candidate that's going to go through the different steps that we might go through and talk a little bit about what makes that such a special plant in the case of legumes, and would it be a good candidate to fly. So Michael, why don't I give you the floor here and talk a little bit about extraterrestrial trifolium.
Yes, it's for a trifolium potentiation. So there's a lot of things we've got to consider when we're thinking about a space plant. And some of the things, the up mass is really critical, the nutrition, psycho-emotional needs of astronauts, space for cultivation, sustainability. And one possibility is clover. In terms of, you might be saying, well, but you can't really get much nutrition out of trifolium, out of clover. But this can function as your pioneer crop that would help establish a soil for very little up mass that you could then use to grow other high nutrient legumes. The clover, literally the trifolium means 3 leaves. And most of the legumes, when the first leaf that comes out is a singlet in a pair that are opposite, and then they alternate the trifoils. And here in North America, they were something that the Native Americans would look for because you had all these flowers with the red clover. And this is actually a place where I kind of got connected to clover. And as I was thinking about all the different legumes that we're looking at and which one would I want to take on as my team, I remembered back to being, I don't know, probably was in 2nd or 3rd grade, and in the spring, being outside on those nice days and literally munching on clover blossoms because they just tasted great. And the bees are flying around. It's that is the one piece that we would have to pollinate them, because unlike most of the other legumes, clover and alfalfa require an insect pollination. You can eat leaves and make a green vegetable out of it. The seedlings are actually good as a microgreen. The little sprouts, they're a little spicy, but that's good in microgravity, spicy in a good way. I like spicy things. A little bit like a mustard sprout or a radish sprout. So it was pretty pleasant. There's some indication that there may be some medical applications. Not much solid science on it to prove it, but there is a growing body of evidence that suggests that it may have medicinal uses as well. And there's some possibilities that it may help with cancer treatment. So those would all be good things in terms of our exposure to the cosmic radiation background in space. Having the clover as something that we are eating might help us to be more resistant to the impacts of that. So coming back to that upmass issue. So here's kind of a comparison. If we take 1 gram of seeds, so 1 gram, that's about the mass of a large paperclip. So it's not a very large mass. What's the going rate per kilo of upmass now?
More than I'd like.
Yeah, we're definitely in 5-6 figures with that. So being able to send a gram, 2,500 seeds was my calculation in a gram of clover seeds. Even alfalfa, we think of that as being comparable in terms of size and everything, but the alfalfa seeds are substantially heavier, and you can't even eat alfalfa. You can only eat the sprouts. And that's the closest one. It's 60 seeds per gram. If you're going to send chickpeas or soybeans, you get 3 seeds per gram. So while they're high in nutrition, you're not going to be able to get as many seeds up in those first expeditions. So it's there's a lot of bang for your buck, literally, with clover.
This number of seeds is the most important metric, though, right? It doesn't
Say
matter
again, please.
It surely doesn't matter how many seeds you send up, but how much biomass those seeds turn into.
Exactly. Exactly. And with the
building an argument for clover, though, you know that.
But I'll stop sharing that screen. And okay, can you see my clover plant now?
We can just see your presentation.
Yes.
Okay. Let me get out of. I just got to get back to the right thing. Hang on. Oh,
yeah. And the
oh
reason I
there's
had
share.
the reason I had that interjection is because I have a study I pulled in that compares like yield versus like the weight of the seeds, and so it was from the USDA. So the USDA chose that metric to be more informative,
Yeah.
as opposed to number of seeds per gram. But
Right.
so
No,
yeah.
but you do one of the connections that I was wanting to make with that is you do get a pretty high biomass per seed with clover. These are mature clover plants. They haven't even bloomed yet, and they are about 20 centimeters from tip of leaf to the end of the roots. More than half of that being in the root mass. So you've got a lot of surface area for potential nodulation. And here I've got, this is one of those seedlings so that you can kind of see how much of this is root mass. And I just, when I was with my fourth graders, there's a lot of nodules in here already, but there's one that's particularly large that I wanted to share with you, that's where the whole thing is happening. That's where the nitrogen fixation occurs with the symbiosis of the organisms. I'm just going to find my right image here.
Is that growing out of rock wool?
It's just rock wool at the top to hold it, and then it's just water below with some osmocote in it, just to keep the roots clean for nodulation observations. Yeah, so I'm going to stop that share and find the image that I want. Oh, here we go. So I just literally captured this image a half an hour ago from the plant that we were just looking at, that I had taped onto that board. Trying to get back to Zoom to share. Sorry. Here we go.
Huh.
All right. So that is a root nodule.
Ooh.
And it is about 3 millimeters long, about 3 millimeters long, and about 1 millimeter in diameter. So I have not done a cross section of this one, but it is definitely, I mean, you can actually see it through here, potentially some of the leg hemoglobin, the pink that's showing through the root tissue. So this is the outside of this encapsulating the bacterium is actually a root hair that was infected by the bacteria, and it's gone through this whole process of changing the physiology and the functionality of these cells, so that now they're working in symbiosis with each other. We've got the rhizobium is taking the nitrogen from the air and turning it into the nitrogen compounds that the plant can use, and the plant is providing it with the sugars, the glucose that it needs. And when we do the cross section, I'm going to find.
Hey, Michael, there's a question. Yeah. What's osmocote?
Oh yeah. Oh, osmocote. Oh, here's the root hairs. Osmocote is their little. It's a particular brand of fertilizer. I think it's 10-10-10 nitrogen, potassium, phosphorus.
And it's slow release. That's what's
slow
so
release
about it.
Right. And I like that is what I use for feeding my plants because of that slow release. If you get a nice steady couple of pellets per about 5 grams of your potting media or so, seems to be a pretty good average. But these little tiny root hairs, this is what gets infected. So this is actually a piece of clover root. Looking at the little hairs that extend from it.
What's the magnification on that?
This is I'd have to do the calculation because that's through the camera. It's magnified even more, but it's this is probably about 600x. I'm gonna just do so I can find the one I'm looking for.
Oh, I love. They look so alien when you see them
Right,
laid out
don't
like
they?
that.
I love it.
Where is the image I'm looking for? Oh, there we go. Okay, so this is the image so this is the cross section of
Make sure
another
you share
clover
it.
knot. You're not seeing that one?
You gotta
Okay.
share. Oh, I love microscopy. It's so cool.
Yeah, I've really been enjoying it, getting back to it. It's been like forever since I did anything of any substance, and so it's the last couple of weeks. It's been great to get back into it, and it just combines, my photography love and just exploration. So this is a thin cross section of one of the nodules from a clover plant. I don't think it was the same one, but the blue. So this is methylene blue stain. This particular image is around 400 x, and
Why did you use that particular stain?
Because of this. The root tissue takes the blue, and you get the beautiful blue. But I think it's a reaction with the leg hemoglobin. I'm not positive, but the undifferentiated cells that the bacteria are in, it gets kind of a greenish hue. So you get a real distinction between the two cell types. And so you've got coming down from center top to going counterclockwise, that's actually the root that wraps around the side of the green blob and then extends down. So I just clipped the ends of that and then did my cross section. And then when you look going clockwise from about 10 o'clock around, going clockwise, that blue is the root hair that became the outside of the nodule. And then you've got this green area in here where all of the nitrogen fixation was happening. So you've got that exchange of nutrients going back and forth between the two organisms. So I find this particular image a really exciting one, just because of everything that I know is going on in here. Plus, I think it's just kind of a beautiful image. I was trying to think if there was any.
This is what we call a representative image. So,
yes.
What I mean by that, well, when you've got a theory about what something's going to look like under the microscope, you go there, and there's a long period of trial and error. This is the artistic period where you practice, you're making your lab book, so it's reproducible. But then there comes that moment when the pattern that you see in the microscope looks like how you imagined it previously, based on the science and theory that you'd constructed thanks to your peers and teachers and such.
Exactly.
And then that's the one you show everyone, your representative image.
Right.
It is representative of your experience as a whole, but we know there's been some trial and error to get there.
Yeah. No, this was not the first. This was probably, I don't know, maybe the 100th cross section that was this beautiful. There were others that were. I mean, I could look at it and see what was going on, but it wasn't, as Richard said, it's not one that I would share publicly.
And all good scientists have these massive databases of old images that they've acquired where something didn't quite look right. Something didn't quite represent the pattern that they knew was there if they could get it just right. And this is why we design experiments and do these iterations over and over. And this is why microscopy is so exciting, because it is really difficult, because there's so many variables before that moment that you had to get right just to get the nodule to form. And then when you've got that nodule, which dye do you use? There's so many. You go into Google, you go into the internet, there's so many different ones to choose from. But there is one which has that right chemical reaction with something in those cells that you know is biologically insightful. And this one has something to do with nitrogen fixation. It's probably something about the nitrogenase in there that's interacting with the dye itself. Yeah. And then you just get this beautiful artistic product that looks so wonderful, and plus also means so much. And can mean so much more when you describe to people the methods that you use to produce it, because that helps them interpret it in the way that you are too. It really is exquisite.
Well said, thank you.
Also, it looks just like an eye.
I'm sorry, again, please.
It looks a bit like an eye of a newt as well.
Yeah, it does, doesn't it? So, and just to remind everybody, this is what we were looking at. These are the nodules the roots. And you know, you can see this was a very productive plant. There's a lot of nodulation on here, and most of it I'm seeing, it tends to be up near the junction between the root and the hypocotyl. That's it, just below the soil level seems to be where most of the nodulation is, though I have found them further down in the roots. But generally in that first few inches or few centimeters of the soil surface, if you're digging things up outside, and the nodules can get really big. Like soybean nodules are several times this size.
It's almost like they require a signal to come down from the shoots to give them permission to fully swell.
Yeah.
So referring back to last week, like
exactly.
Sugars, right? This is what we were talking about.
Yeah, exactly. Your casual observation here about the increased density of nodule formation at the top of the root, like near the base of the shoot, in itself is again what makes like microscopy so beautiful, because there's these moments where you see something, you think it's a pattern, but then you have to do the microscopy again and again to see it some more. But like then you can take that pattern that you've seen, i.e. the story of the clustering of the nodules down near the base of the top of the root, the base of the shoot, and then we can link that back to the science that we've read about and discussed previously, and postulate a theory. And that theory may be that these nodules form there because that's one of the locations they can get a message back from the shoots after the roots send a message to the shoots. So again, I'm just bringing in a theory here based on your observation, linking it to some data that we saw in the literature previously.
Yeah,
and this is what makes
this was also an adzuki. This was not a clover. I'm just looking at my notes. This was a 20-day-old plant. It had just formed its third node, so the second trifoil, and it had 54 nodules. And I had a mean average of about 3 millimeter diameter. So this was
nodules per gram, I get, or what's what's the percentage of mass to the nodules, right?
Right.
So you might have something that's very abundant in terms of seed count, but nodule mass, I guess, would be a consideration with respect to this particular experiment we're trying.
Exactly, and I can how do I unshare? Oh, here, stop share. So yeah, and that's the piece where I'm focusing on clover now. I don't know how well you can see my legume garden over here with the bright magenta light, but I've got our original cowpeas. I've got adzuki, mung bean, chickpea, a couple of species of clover, alfalfa. What else have I got? I think that's about all of them that are there right now. And I'm now focusing in on the clover because I'm liking that particular candidate for this experiment. And I've got a new variety of clover that I'm trying. That's a dwarf variety. So I'm going to be doing the work on that, and just exactly what Ted and Richard are saying, really establishing how much biomass am I getting from per gram of seed, as well as the nodulation potential. And then one of the things that we need everybody to figure out is, so I'm seeing around second or third node is when the nodulation begins to occur.
How many days?
Well, it depends on the plant. Yeah, it's about, the adzuki was a 20-day-old plant. Mm-hmm. Clover isn't that old when it gets to its second or third node.
Diana is one of our PIs and you've got glycine max soybean,
right?
And actually, I think he called soybean out in terms of you're a heavyweight bean, right?
Heavyweight,
bean-tastic. And so, but you also said, Michael, that those nodules were pretty prodigious as well.
Yeah, it can be. Just remember, originally I was, long time ago in a planet far, far away, I was an Iowa farm boy. And I remember that's really when I got fascinated with legumes, was just how could these plants produce roots like this that had warts all over them, and some of them pretty big. What was going on with that? And then just coming to appreciate all of this incredible biology that's going on there. And there are lots of, as as Diana will tell us about glycine max, there are a lot of applications for it beyond just a food source. So
equal opportunity, Diana, if you want to just weigh in a little bit with respect to your hero.
But I don't think her hero can do it on its own. I think it's going to have a pioneer plant to establish it.
I don't know. I'm telling you, I think that I'm not sure what beans that you did send to us, but as I mentioned to you that I did get a separate bag of beans here. And
yeah, yours are
we got
black.
some real growing going on.
Yeah.
Growing. I'll tell you very quickly about the resuscitation that was done after taking it out of the polyacrylamide crystals, because I thought that plant was done. It was also one of the rhizobium infected plants. And so I took it, moved it into tea, because that's what I'm working with in most cases. So the tea grinds and a little bit of Miracle Grow earth. And then I squirted it with a mix of water and peroxide, and we've got some growing going on. It has completely recovered, which is amazing. Good. The other thing too is somebody mentioned earlier about light. This doesn't need a lot of light. It's really just been hanging out in the darker part of the dining room here, and it's been doing very well. So from the original batch, I still have some growing. So in the original batch, just turn that around. Just get you to the right one here. That's a zucchini.
You know, and the light too. And one of the things that I meant to comment on with that question was, we are also going to be we're going to be using artificial light, because the person that asked the question, you're absolutely right. There's not enough intensity from the sun to get very high production from our crops on Mars. So we are going to be having to supplement with artificial light.
So they're growing crazy. I've got them in the paper towers that I created. And these are also rhizobium infected. So I'll be pulling them out really soon to see how they're doing, because they're getting very, 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 very large. I'm gonna give this one a bit of a hand in a second. And then the other thing is that with the other beans that I purchased, all right, they're sitting in a bed of earth, rocks, which is just aquarium rock on the bottom. And then on top of that, cedar chips. And I mentioned last time that I literally warmed every bit of it except the rocks in the microwave. So the rocks were sterilized, but the microwave is what kind of cooked our earth. And they are
like Mars.
Yeah, I'm going for Mars. They're growing really long. They originally, after just 2 days of being in a plastic bag, and this time I kept them off the window because we're still going below zero at night here. I kept them off the window, and they're doing really, really well. So the bags were just very simply small Ziploc bags, and inside was a small piece of actually paper towel that had been sterilized. And I squirted it with the peroxide and water. And so far, every time I hit these things with a little bit of peroxide and water, they go boom, and they keep stretching up. So this is just as of last night. They're not taking long to grow. That's the other thing. The peroxide and water in the Ziploc bag with the beans in it, they showed roots right away. And that was within less than a day and a half. So that was really quite a surprise, because the other ones didn't come out that fast. They took a lot longer to come out. And then I've got some other things that I'm trying, like the adzuki. The adzuki's doing well. Lori and I were talking about putting baking soda, and I tried the baking soda, and I did a baking soda base. And so that seemed to be doing okay. But then I looked closely at my plant the other day, and one of them was just wilting and going down. I thought, what's happening here? Because the other one looked fine. Well, I hadn't realized that as I was dusting a little bit of that baking soda across the top of the earth, right, my medium, I actually hit the plant. So inside this little bucket was this baking soda that was just completely draining the plant. So resuscitation too now is coming along, because I've been feeding that one with not the baking soda, sorry, I spilled some water, but the peroxide. Peroxide seems to be boosting all of the plants, even when they're looking kind of shaky here.
Richard, there's something going on there chemically and physiologically, right?
Yeah, hydrogen peroxide is absolutely fascinating.
It is.
It turns out if you take some of the space-flight studies and you look at the genetics of the plants responding to spaceflight in a dark environment, and you compare that to studies on Earth, the most similar one is the addition of hydrogen peroxide. I don't know. But in this context though, hydrogen peroxide, when you're adding that, you're activating the ROS. You activate, you know, those calcium waves that I'm showing? Like they require the production of hydrogen peroxide in the apoplast. That's the space between the cells. So when you're adding that hydrogen peroxide, you're activating this electrical wave that's going through the plant. That is part of the signal that causes the microphiles to open. The microphile is the pore at the end of the seed. So the pore at the end of the seed is where the water gets in. As the water starts to get in a microphile, it washes away the hormone that promotes inhibition, like APA, the stress hormone. It then allows, that water then allows the production of germination hormones like gibberellin, which then starts to activate ROS production in the apoplast outside their cells. In the elongation zone, that causes cells to stretch, so the radicle starts to push its way out of the seed. So basically, by adding that low-level hydrogen peroxide early on, you have given a kickstart to the germination process.
And see, I'm not a scientist, I'm a teacher.
So you are a scientist. We're making a scientist out of you.
That's right. I love discovery like that, where something happens and you back into it. That's what it's all about. How many more discoveries can we find? And even if it's like been written in textbooks and well practiced, it's a discovery if you've never experienced it before, right? So it doesn't matter if you're 14 or 44.
Yeah, Carl Sagan said, even if you are the very last person to see the light, you will never, ever forget it. And that's the power of discovery. Even if it's been discovered 8 billion times before you, that 8 billion first is yours. And that's the one that will stick with you forever.
And for the teachers and the kids that are out there, this whole thing is about exploration. And Ted, when you started today, you said something, you said, just grow plants. So because you said that, something triggered in my brain, and I just started mapping out a cover. So now I've got a cover that's going to go with the lesson plans to the students for this summer. And thank you, Ted, because we have now just grow plants. So we've got a bilingual cover just from what you're talking about. And I think that this whole exploration phase is really key for us to make sure that the teachers and the kids grab onto that, because this is where they're going to realize it doesn't matter what you try, it doesn't matter what you do, you're going to get results one way or another. And that's the fun part of it. It's either going to be great or it's going to be bad.
But you always learn something in the process. And
that's right.
That's what science is about. It's not proving.
I think you froze.
But it's what are you learn? It's the journey. That's what it's all about.
Yeah. Javier,
Mike, just very quickly to call you out. Okay. Glycine Max.
Trifolium pratense.
Now I notice Javier and Lori are on. I don't know if you guys have selected a candidate or your principal investigator on one. Of course,
I think they're being Leguminaut agnostic.
Well, I am too.
I'm doing cowpeas. I think there was no one assigned to it, so I'm gonna be doing the cowpeas. But I wanted to share a little bit of what I've been kind of observing with medium. So this is just a paper towel, and I wanted to see how resistant a plant is without any medium. So I've been trying to research this. I can see a little bit of mold. This is like a 3 or 4 week, lentil. that I was experimenting with my kids. So we finished with our activities, but this still growing. So I'm impressed that with just a little bit of water and without any medium, is still growing, and has been like this since, I don't know, long time. But I'm also comparing and contrasting, and one of the inspirations for this was Michael, because teachers try to get everything perfect, especially for science and stuff. But then we went to a training in February, and he was mentioning his journey as a teacher, and then he told us, hey, why don't you try it? It doesn't matter if you mess it up. So I'm taking his words for granted. So now I'm experimenting with aloe vera, and I am comparing and contrasting. There's a little bit of light here. Comparing and contrasting just with cotton balls and aloe vera. According to some research, aloe vera is 99% water. You can see a little some difference with the lentils. These lentils are a little bit smaller, but I'm documenting everything. They look smaller than the cotton ball, but there's another plant. I think this is cowpeas, or no, it's mung bean. I think
it looks like mung. Yeah.
It was growing faster, but just suddenly yesterday these began to sprout. So it was pretty interesting. But one of the seeds, it's still small. These two are kind of big, and this one, you can see the roots is kind of semi-transparent. It has a gelish consistency. And this plant is bigger, it's mung bean. And this is the contrasting one. So I'm interested and intrigued to discover if aloe vera might be a good medium for the plants to grow, especially since they, according to what I've been researching, it has some components that kill bacteria, but I don't know if that's gonna kill our friend bacteria. But I'm not sure what's gonna happen with this. I'm been observing a lot of stuff here, the colorant, pigments and stuff, and has a lot of properties too. So I'm interested and intrigued what's gonna happen in a few days.
Well, yeah, me too. I'm gonna have to bounce to my fifth graders now, but it's been great hanging out with everybody. We'll talk to you again soon, and keep up the good science, everyone.
Mm-hmm.
Thanks, Mike.
Yes, and I've been trying to research if there's another people trying to grow seeds from aloe vera, but I haven't find anyone. So I'm gonna be the first one, I guess.
Well, I can give it a shot.
That's really cool. Is there, what are you using though? Is this a liquid? Is it a powder? Is it from another plant? Like, what is it?
Just the leaf. It's just a cross section of the leaf. I use the tube as a template. I just mark. I push it a little bit and begin to cut around, and I just stick the little plant. You can see a little bit of the green and the gelish or the gel here.
Thank you.
Interesting.
So it's interesting.
It's kind of like grafting, isn't it? Like, I know that in some industries they're like slow growing mushrooms, and so they graft the mushroom from a slow growing plant onto cacti, from a slow growing cacti to a fast growing one, and to make them grow faster. They do similar things with other plants. It's like you're using the aloe vera here as the stock, and you've got your new plant growing from that. That's really cool.
So I don't know what's going to happen. So we need to wait and see.
That's very interesting. And Javier, while we still have you there, you're doing some interesting things with university students and also younger kids as well.
Yes. So we are doing the Leguminaut challenge as well, but the kids are going to be doing some research on how to do the habitats for the plants, or basically a space garden. So the challenge for them is to, with another teacher that is leading these students. His name is Professor Max. He's in Mexico, and they are working with, I think it's 12th graders. So they are from, not just from biology, their background is in computer science. So they are using their computer science and mixing it with the Leguminaut challenge, and they are going to produce some virtual reality models of their garden. So it's going to be pretty interesting what these students are going to be developing. They have also a previous experience doing these habitats, but they were doing PVC pipes and plastic and stuff. So I was telling them that this is going to be something interesting because they don't have any restrictions based on the malleability of materials, or probably you have certain meters of PVC and that's it. With virtual reality, you can model, you can scale it, and you can even go inside of the model. So that's a program that we're going to be working with these students, and I'm hoping that it's going to be pretty interesting. And these models are also going to be, it's not the competition with the SciArt. They're going to be just showing up their productions there. Mm-hmm. So yeah, and one of the things that I was talking to them the other day was the same thing that Dr. Barker was telling us in his journey, that, you know, his hobbies are now part of his journey. I told him that in Mexico we live in a place that is very enriching, and they have a lot of things that they are looking around. They have a lot of hobbies. They are enriching themselves with vocabulary, with foods, with methods that we use for surviving. So I told them that one day when they are in the field industry, they're going to use their skills and everything that has been surrounding them. They're going to be pushing that in their careers as a professional, because what we discover is not just by being a biologist or a scientist or an engineer, is whatever is your background that is nurturing you, that's going to be developing the new stuff that we're going to be needing in the future.
It's so wonderful to hear you say that, Javier. One, I guess this is a great point to make. We have the scaffolding, which is saying, help us select a legume to go to space. And we have this 1-year program, and we're going to send this up there, and we're going to be doing some research. But there are so many different avenues of exploration. As an educator, if you're watching us and you're thinking, how can I fit this into my program? I'm an English teacher or whatever. There are so many ways to connect these points. Feel free to reach out to our team at Magnitude, just hello@Magnitude.io. Share with us your kind of pedagogical format, where you're trying to take your students, whether they're in primary school, whether they're university students, and let's collaborate. Let's find a way where we can engage them where it's meaningful. Here we've got computer science students working with virtual reality that are now involved kind of on the edge of what we're doing. We're just trying to nodulate some legumes just. You know, it's pretty fascinating to be able to pull that off. But now we have other groups that are joining us because they're taking this and extending the thinking. Now, Lori, if I can, I'm going to kind of switch back to thinking about our challenge. I just want to bring that up. But Lori, I guess you have some students that are just finishing up the school year, and you've got some results you might want to share with us.
Yes, so we'll see if I can share my screen with you. My students have been working on this Leguminaut challenge for the time that we've been in the quarantine pandemic. And so they've submitted some final projects, and some of them are just amazing. I have not picked my ideal legume to fly, so I'm in the in-between stages. And some of my students' work could definitely influence the choices. So let me see if I can share here my screen. So you'll tell me it shows up. Can you see this poster?
Let's see.
Yes.
Looks like Google Classroom, and now we see it.
Do you see a slide hopefully starting to appear?
It's coming. It says loading.
It's pretty graphic intensive.
It's very deep. Yep.
They created these large posters. Since we couldn't create a poster in the classroom, we did Google Slides, and created a massive poster. So if you printed this out, it'd be about, 3 feet.
Wow.
So do you see it yet?
No, yeah, we see it.
You see it? Okay. So this was Ruby, who was on with us just a few weeks ago as one of our students.
Mm-hmm.
And so they had to do research about rhizobia bacteria and the relationship with their plants, their chosen legume, to look up what does a root nodule even look like. They had to look at, as the seedlings were growing, and integrate pictures, find recipes to connect. How can you eat these different legumes, as well as going deep into the nutritional aspects of legumes. As a biology teacher, I really tried to apply their learning this year to real world application, and for them to understand what they put in their bodies, and it either is going to help their body function, or it's going to, in some cases, be detrimental and harmful. And so looking at the nutritional aspect of these legumes is important, because when our astronauts go to Mars, they will need nutrient-dense food. And sending prepackaged foods, those are going to degrade and lose nutrients. Therefore, we want to send the ones that have that highest nutrient density possible. So they looked at the different vitamins that are crucial that are found in each of the legumes. Here in the center, I wish you could see this bigger, but here in the middle, she did this beautiful watercolor. It was a pencil and then water color kind of finish on top of it of the lentil development. And then finally this week, we ran a salinity test on the different legumes and compared how they did in a 0.5%, a 1%, and a 2% over about a 7-day period. And the lentils were extremely sensitive to the salt almost immediately. Most of them, the growth was just completely stunted, and they started dying off. You could tell the roots were shriveling up. So that's one of them. Let me see if I've got one more.
And if you can share those on the drive or something else like that, that'd just be wonderful. We'd love to kind of.
Absolutely. Absolutely. Let's see if I can present this one. This one was on chickpeas. And so I like how he did his background and found the different images of the root nodules, put in the table with the salinity test as well. And it was interesting that the chickpea and the garden pea were the two that were most tolerant to the salt test. There was some shriveling and browning of the roots by the last day. Their growth wasn't as strong as the control group, but they definitely tolerated the salt a bit better. So, and you can see here in the bottom picture in the Petri dishes that if I didn't tell you which one was the control group versus the salt group, you wouldn't have been able to tell on this one as much compared to some of the other legumes. But they just finished these projects and turned them in. They did a fabulous job. They were required to do some extensive research in finding others, experts who've looked into these fields pretty deeply, and to really process that information. So yeah, it's pretty exciting. A number of them have given me feedback on their exit ticket for the course this year. And they said quite a few of them have already noted that the Leguminaut Challenge was their favorite project of the year. So that really made me feel good that during this pandemic, I got them involved in something that was memorable, was impactful, was a positive experience for them. And so many more of them were also saying that they want to learn even more about plants. So that is so exciting, that their curiosity is piqued, and that they want to find out what happens. They want to stay involved. A few of them have signed up to take my AP Environmental Science class next year. So of course I'll continue their training and learning all about legumes and putting them in space.
How awesome
That
is
is
that?
so awesome to hear. I would love to be able to interview them.
Yeah, so that would be a good interview with some. I'll see if we can get that arranged.
And actually, if they'd like, we have 2 sessions remaining. We might have a slot available if one of them or a couple of them want to present their poster in more detail firsthand. We welcome them to do that. And actually, any students out there, I'm just going to throw it out. If you've got students, whether you're in the Zoom or you're on YouTube and you're part of the Leguminaut Challenge, let's showcase your students' work. Students, if you've got something you'd like to share, we'd love to hear it as well. So it's really about sharing knowledge and working through these experiences together. It's always a lot of fun.
Absolutely. It's a wonderful way to engage students right now when it's so challenging with trying to wrap up this school year and coming up with lesson plans. This is, I'll be happy to share my plans with other teachers in making this happen, and the different segments of that we did, from nutrition to the rhizobia research, coming up with the.
Uh-oh.
I was going to ask about the recipe. There was a recipe on the poster, like a link to it, and that'd be such a good thing in the class too. Like, it's one thing doing the research and the science, like the health benefits from eating these things is so good, and so to have like a hummus like bake off in class, is so good.
There you go. Actually, that's a great segue. At the top of the hour, we're supposed to be joined by Leguminaut, the restaurant there in Orlando, which focuses on legumes and all the recipes. But Beth had, it sounds like a little family emergency today, so she's dealing with that. But she did forward a little video of her recipe of the day. She's working with mung bean, and there's a company out there that takes mung bean and makes an egg. Well, something tastes just like eggs, I guess. So it's just a short little 2-minute video, but I see Sarah's here from University of Florida. So hey, Sarah, I don't know if you started working on the program at all, the challenge, or if you've got some of your future farmers of America. You want to share a little bit about that, what you're doing there?
Yeah, so I'm trying to get connected right now to high schoolers. It's difficult because of the timing. I'm working on a lot of stuff, and they are too. They have their finals coming up, and their like FFA advisor is also getting married. So
oh wow! Well, congratulations to them.
Yeah, it's really good.
So but one thing, I guess I can offer is, we'll be running this for an entire year, and we're just in the first month or 2 of it. And so if you want to get these young folks involved during the summer months, really kind of looking at you as really one of our co-investigators here, would love to find a way to work with you directly and getting that underway. It may be in preparation for the fall. So just guide us and let us know what you might want to do there. That's pretty exciting stuff.
Yeah, I will. Thank you. I'm looking at getting a smaller number of students, maybe like 5 or 10, hopefully. So not like a full classroom, but more like a focused group of students that are interested or maybe interested in plants.
Oh, that sounds fantastic. I think with the pandemic, that might be the size of a modern classroom. Right? 5 to 10 students. Well, that's wonderful. Even an additional person, bring a friend along. It's really exciting that you're excited about it. Share a little bit more about what you think about the challenge, and have you started growing any legumes at all yourself? I know you're getting finals your last week or 2, right, of school?
Actually, my last week of school was a week or so ago, and my new semester has started.
Oh, wow.
And I'm taking a gardening course, so soon I will be growing legumes. So I'm like gearing up for that and getting the materials I need and everything.
Well, fantastic. Now, for everyone that doesn't know, if you're active in the challenge, on Wednesdays, 3:00 Pacific time, we have a 1-hour session in which we just kind of get together and kind of share the state of things. So, you're definitely welcome to join us and I think you might have been on one of these sessions before, but I'd like to see how your gardening's coming along. It'll be a lot of fun.
Thank you.
Let me just open it up to anyone here that wants to share what they're up to. Otherwise, I'm going to play the Leguminaut short little video. It's only like 2 minutes. She's going to introduce the egg. See if I can find.
I want to share a little bit of my my class. Let me just piggyback writing with Lori. Let me share my screen.
Okay.
Hey, Javier, I really love your video in the background.
Oh, yeah. Cool. You see the screen, guys?
Yep.
Okay. It's not very high resolution, but this is the version of Lori's. This is a student that is very meticulous. Mm-hmm. And she, whenever we begin the Leguminaut challenge, we set our rules. This is a work from a kindergartner perspective. And what she did was the whole process. We have a lot of challenges because since they are new to gardening, to growing stuff, there are things that has to be preview before teaching, or some stuff that has to be reteach before moving to new stuff. And as Ted was saying a few minutes ago, that not just because you are in a different field, this doesn't pertain to you. So this is a class that was, the main focus was about language and writing skills. So I developed this thing about those two items. And what she did was incredible. She began to record the growth of the plants. She did also a simulator of the plants. So this is what she was able to get from the simulator. This is a little bit about the process, how she planted the seeds. A few days ago, after planting, this was a drawing, the parts of the plants. She also did the parts of the seed. She did the dissection of the seed with the help of the parent. And this thing is very interesting for them because they were able to compare and contrast two plants. And this is one of the most important things and skills that a kindergarten has to have, comparing and contrasting. And, you know, as we mentioned a lot with Dr. Barker and everybody, that you need to notice the differences between plants, and that's a very important skills for research. And they're doing that from the beginning of their school year. And this is a plant that they were comparing with sunlight and without sunlight. And she drew the parts how the plant grew, and she did a little bit of writing on the observations. She even measured. This is 8 inches and this is 6 inches. So she did all of that in kindergarten. So
oh my goodness,
imagine how amazing job I don't know, a first grader, 5th grader can do. This is just kindergarten.
I love this spectrum here. We see high school, juniors and seniors in high school. We see someone that's 5 or 6 years old. Everything in between is possible, even for old guys like me. Jump in, do some art. There's a couple questions that popped in here while we've been chatting. Let's see. We've got the color of the stem. Fernanda asks the question: the color of the stem and the roots of the plants, can they determine the properties of the plant? I don't know if anyone wants to weigh in on that. So I guess Dr. Barker hits the buzzer.
Ding. Oh, it's funny you should say that. What was the question again? It was.
It's about plants, for sure, right? So the color of the stem and the roots of the plants. Just by looking at the stem, can that help understand the properties, like maybe the chemistry or what have you? Okay, cool. Chlorophyll expression, I guess, is that indirect question or?
Well, it's such a good question. And do you know why I like questions like that? Because it links to a data set I've already processed. I'm now loading up.
Right, great.
So stems and roots. Earlier, not long ago, as part of the Leguminaut challenge, Michael gave me a challenge or call, and he said, "Hey, I see some pigment in my root. What is this pigment? Could it be anthocyanins?" And I was like, "Well, maybe." So I went off on a bit of a tangent looking at pigment. So I pulled in a few papers from the USDA, US Department of Agriculture, to look for studies about pigments and nutrients. And basically you can link phytochemicals, so these to human disease. So we'll start from the disease, moving back to phytochemicals, then move from phytochemicals into colors and plants. So because people, and particularly NASA, are very like human-centric. So when humans go into space, there's lots more radiation from the sun, being caught in the Van Allen belts, and there's all these galactic cosmic rays and all these weird magnetic fields. So we're concerned about humans and their long-term potential to survive in these extraterrestrial environments because of what's collectively known as oxidative damage caused by these different types of radiation. And this little table down here is a bit innocuous, but what it is, it's a summary of many different research papers. These numbers here refers to research papers in the literature. Scientists like to reference stuff so we know it's real when we say stuff. And what these research papers have done is linked these diseases such as cancer and obesity to phytochemicals that they know can provide resistance to that, like flavonoids and carotenoids and vitamin C, E, etc. And so, okay, so that's great. Okay, we know there's chemicals in our food that can be used to prevent the occurrences of diseases, and some of those diseases are likely to be more prevalent in extraterrestrial environments. So which plants do we want to eat in order to maximize these nutrients? And the question was color. Can we look at the color of the stem and know something about the function in the plant? See, so these chemicals are inherently colorful. And so one of the studies looked at the color of the plants. I'm trying to remember which one that was. No, this is young versus old plants. Nutrients change as plants get older over time. So eating younger plants is more nutrients than eating old ones. Color, this is not the color, this is the yield page. I'm terribly sorry, it's been a while since I've been in here. This was yield versus seed size. Keep going. Aha, here we go. Yes, quite. So, okay, so a moment ago, I was raving on, okay? I was trying to persuade you guys that there's nutrients and there's disease, and that some nutrients help reduce the prevalence of certain diseases. And so if we eat the right things, we can give ourselves nutraceuticals, nutrients that will prevent us needing pharmaceuticals. Okay, cool. So here we have a whole bunch of different types of organisms that we could eat. Peas being the only legume in the list. Basil, I love basil. Yes, you're pronouncing it wrong, not me. And so here we're just looking at the dry weight. And what we can do, we can change that to different types of nutrients that are in these in these different species. TAA is one. Oh, cabbage, that's my favorite. Lutein, this one's known to be good for protecting your eyes, which is a real issue for astronauts. Okay, cool, so we've got different types of nutrients. We were talking about previously about certain nutrients important for health, but the question was pigment. Can we look at the pigment of a stem or root and know something about the function in the plant? Okay, cool, so those chemicals we're looking at, those nutrients, well, they're antioxidants. We know that we want to eat them because of their role in disease. Okay, so now what we've done, we've grouped the plants based on their color. So we have some plants that have been grouped collectively. Okay, so here are all the different plants we have. We can group plants based on their families, based on the individual species. We can group them based on like commercial summaries, like grouping together all the radishes or all the mustards, right? All the beets. But then essentially what we can do is we can then group these different plants based on the color that they become, just based on photography. So some are green, some are purplish green, etc. Reddish green. So I'm going to go for reddish green and purplish green and purplish green and red because it's got red in it. I'm just going to scroll down a little bit. Okay, cool. So what do we have here? Now what I've gone and done is I've linked the colors. So here we can see I've put the filter to get rid of just the green plants because they don't have as many nutrients in them. They're still good for you, but the colorful ones are better. And so what we can do now, we can see all these different chemicals here. So some of these chemicals here have been found in plants that are known to be purplishly green. So what I was trying to get back to is to the student's question, which was, if we see pigment, if we see color in the plants, can it tell us something about the function? Yes. If we start to see like a purplish, greenish tint occurring, that means the plant is producing these chemicals, these secondary metabolites, anthocyanins, phytoquinones, some other words I can't pronounce, beta carotene, right? And these chemicals here, we know some of those are linked directly to the disease, and so because of their antioxidant like characteristics. So it almost answers the question, but it has enough science around it to make it seem like it did even out. What was
Well, it probably opens up additional investigation now, doesn't it?
It always does. And that is the sign of a good question and of a good research project. People think by the end of a research project, the problem will be solved. If they phrase the question in the right way, it will be answered easily. No. Good questions require a proper research project to try and uncover the answer. And a good research project may uncover the answer, but will ask more questions at the end than it answered at the beginning. So that's the fun world.
If I can add, so the student could experiment this summer with carrots. Okay, so there is an experiment from Kennedy Space Center that looks at chlorophyll. Put your carrot seeds like in a water bottle and put one seed close to the edge of the bottle so that it gets light. Plant the other seed on the other side where it's dark. And then tell me at the end of the summer, what color are your carrots?
Wow, I'm gonna go do that.
Because the light will affect the color of the plant. So why are carrots orange? Can you come up with a carrot of a different color just based on the light?
So I've got some rainbow carrots. my friend, she was responsible for the sequencing of the carrot genome. So she's a really awesome, inspirational woman, just like you. And one of the things that she does is grows carrots, and she dries them out and makes jewelry out of them, because she's been collecting lots of different variety of carrots. So she's got some purple ones, she's got some white ones, there's orange, and there's some that change color as they go through. It's amazing. There's such diversity in carrots.
Wow. That's, I guess, a good sign of a great question. We've just opened up this whole other avenue of exploration. See, there's another question here too. James asks, so little bacteria to a lot of plant. How much nitrogen is produced? Any way to quantify the amount of nitrogen produced?
That's hard. So by nitrogen produced, he's probably referring to the nitrates or that have been absorbed and made biologically available, because you're not producing nitrogen.
Right.
Nitrogen, it's just there, you're changing its form. You'd have to do it with radioactively labeled nitrogen. That's how they do it in the labs. So you can get different isotopes which have extra neutrons in their like atoms, which makes them radioactive. Not like, oh, glowing, I'm going to die radioactive, but like detectable with radioactive detectors. And so people that do like gas flux experiments, they get radioactively tagged oxygen and nitrogen, and then they can watch its flux through the plant, so they would then start to harvest their roots and shoots at different stages, and then measure the radioactive atoms that have accumulated inside them. So it's not the sort of thing I like to do. I try to avoid the radioactive work, to be honest. Even when it's safe, you do the protocol correctly, it's fine. It's just, yeah.
Well, in addition to that, on a relative scale, you can know if there's not enough nitrogen by the color of the leaves, right? They're going to turn more yellow. And then if you have too much nitrogen, often plants won't flower. And so I'm even having that issue with some of my tomato plants right now, where I think the nitrogen content is too high. So on a relative scale, you can tell if there's just the right amount.
Hit them with a stick, your tomatoes. Give them a shake. A little bit in the morning, a little bit in the middle of the day, and a little bit in the evening. If you give them a regular shake, it makes them flower earlier.
What? No way.
Yeah, like, so to test this, we built a robot. We're just in the process of putting the paper out that tickled the plants regularly with like a plastic glove.
Ticklebot.
Yeah, because we found humans aren't like reproducible enough. Like we would shake it or hit the plant at different frequencies each day because we have different things to do every day. But we had this theory that if you did mechanically stimulate it every day, it would cause them to flower earlier, and it did. So go beat up your tomatoes.
Mhm.
Give them a shake.
Oh, that's wonderful.
The 50 grades of the tomatoes.
Yeah, just not enough to crush them. Don't beat them up that much.
You don't want catsup. Hey, so we have a question from a third grader coming in through another channel here. It's just a big general question. So any of our educators here, please jump in. But what types of environment are you going to use to grow legumes? And I'm guessing they're referring to the environment on the space station. I can speak to that, but I want to open it up to anyone else if they want to tackle that first. Otherwise, I can answer that question.
Well, I'm doing just my house because it's regular sunlight and exposure. But I'm also doing the ExoLab whenever I kind of finish with this trial. I'm gonna be moving this to get more data for the ExoLab. So that's the two parts that I'm testing. One is outside, regular sunlight, regular control temperature, and then I'm gonna be switching to the ExoLab.
Fantastic. Now, this question is coming from a third grader. So there are several different components you're going to be thinking about with your plant when it's growing. It's light, right? You're going to need something to create that photosynthesis. You're going to need some food for it. So that's the carbon dioxide that's going to convert to oxygen. It's going to need some water to help move all of these nutrients around. And then it needs some kind of growing medium. Now, Javier actually showed us some plants that are growing just in a damp paper towel. Probably not the most nutritious, but it looked like it was trying to make it work. It's so, but your soil, maybe out there in your backyard or in your garden, could be a great resource for your plants. So think about those things and how you might want to grow it. On the space station, we're using a gel. It's an agar, but we're going to probably change that a little bit because the roots are not getting enough oxygen is what it looks like. So we want to make sure it's a little more like the soil. And agar is really more like Jello. Like that's what Javier is showing you there. And so
agar? It's from Carolina, right?
Yep, that's right.
Brand.
Yeah, and that's a special formulation we created that doesn't have nitrogen in it because we're asking the bacteria to do the work of converting the atmospheric nitrogen so we can use it, the plant can use it. But on the space station, we're going to be in a little box. It's twice the size of this guy here. Just a little aluminum box. We'll have our plant and bacteria growing in it with artificial lighting, LED lights, and it gets a little toasty in there. It gets warmer than what you'd really want a plant to grow in. We're 29C or so. And I think we're improving that to get it down more in the 25 range. Would like to get it even lower, and something more comfortable, t-shirt comfort, not like no shirt, too hot. And so, yeah, it's a little different environment that's growing in. And of course, what's interesting to us is it'll be growing in a microgravity environment. And so there isn't like the gravity you have here on Earth. You jump up, you land back on the ground. If you're on the space station, you'll be essentially floating around. And here's an interesting fact for you that's tuning in and learning about space and about these plants we're going to grow. Why is there and how is there microgravity on board the space station? It's not that far away from us. It's 250 miles above the planet. If you got in a car and drove it, it would just take a couple hours, but you have to kind of go straight up. And there's gravity that's kind of holding us down on the ground. So the International Space Station goes around the Earth, and it goes around the Earth very quickly. In fact, it's traveling at 17,500 miles an hour. That's really fast. It goes all the way around the world every 90 minutes. So if you're an astronaut, you get like 15 sunrises and sunsets every day. Every 40-something minutes, you're getting a sunrise or a sunset. Really pretty amazing. And it's traveling so fast around the Earth, it's essentially falling falling all the time. Like sometimes you get that little feeling in your stomach in an elevator, or if you've ever been on a roller coaster ride, you've gotten over the top and suddenly you're dropping. The astronauts aboard the space station are always falling. They're just traveling so fast. Earth is a nice big sphere, oblate spheroid, a big ball, that it keeps on missing the ground. It's traveling so fast. And so it's falling all the way around the Earth. And as you fall, you have the sense of weightlessness, very small amount of gravity. I think I guess we'd have to get some physics folks involved here, but my understanding of the universe, it's essentially impossible to get zero gravity. Something's pulling you somewhere. But it's going to be very, very tiny amounts unless you're really close to a star, and then it's a lot, or a black hole. That's a whole bunch of gravity going on. In space, as you're on board the International Space Station, you're traveling out to Mars, get a sense of weightlessness, but there is a little bit of tug, a little bit of gravity. It's just that in the case of the space station, it's traveling around the Earth so fast as it's falling, it keeps on missing the planet. It's pretty wild. So that's kind of something about your environment there. If you've got more questions, please ask. Now, a lot of our students that are joining us here are excited about our balloon launches, and we'll be picking those up here in about an hour. Let me share with us, we were supposed to have the Leguminaut folks join us. I don't know if they popped in or not. Let me just check our who's here, because if Beth is here, we can jump in. Oh, I see Jeff. Hey, Jeff from Stockton. Good to see you. Got to catch up, man, to see how you guys are handling shelter in place and distance learning. Always a challenge, right? You got good kids over there, though. So Jeff's class was actually planning on one of these big balloon launches, and they've also been working with us with ExoLab. But let me share my screen. We'll share a quick little video where this company apparently has taken mung bean, and they've been able to make it through some manufacturing process to taste just like eggs. And she's creating a recipe for us, as she does every week. So hopefully, can you guys see that? And looks like this is just less than 3 minutes.
When it's Beth from Leguminaut, I am doing our legume of the week, which is mung beans. And I have this product here called Just Egg. It's a plant-based scramble. It's from this company called Just, which makes plant-based foods that are sustainable and have a lower carbon footprint than their animal-based version. This is their scrambled egg product. You can find it at a lot of popular grocery stores. And as you can see in the ingredients, the second ingredient is mung bean protein isolate. So this is really cool that mung bean protein isolate can become scrambled eggs. I'm gonna saute up some spinach in my frying pan for everyone. Well, I wish it was for everyone. I wish I could make you all some plant-based scrambled egg. So spinach cooks really fast. You can put any veggies that you want in your scramble. You could do this as a scramble, or you could try an omelet. I, the other day, made some with some vegan chorizo sausage in it. It was delicious with avocado on top. So if you're feeling like you want something a little spicy, that is something I would totally recommend. And you can get that vegan chorizo sausage at a lot of grocery stores. Trader Joe's always has it. So I'm gonna go ahead and just like you would pour scrambled eggs, then you're gonna pour your Just egg in, and it cooks. Well, I mean, it's been a long time since I've cooked a chicken's egg, but it's one of those things that you kind of don't forget. So this cooks very similarly in the pan. So you just kind of want to wait for it to kind of stick, and then you can start scrambling it around until you have some really yummy plant-based scrambled eggs. Now, I have shared this product with both vegan and not vegan friends, and they have all enjoyed it. So if you're a little weary or wary, a little wary.
Yep. I think that's it. She, that's all I got from her. But it would have been nice to have Beth with us. Hopefully everything's okay with her and her family. Right. So here's a question I have on the chemistry side: protein isolate. How do you do that? You just can't crush up some mung beans and say, "Look, I got eggs," right?
There's definitely some chemistry that comes with that, right?
Right. And then, you got your plastic bottle and what have you. So some questions there, but I'd be very curious about the process chemically on what you do to make that happen.
It's amazing what you can do with modern food science. When I went to the University of Nottingham, I lived in this little village called Sutton Bonington, and just down the road there was another little village called Quorn. And one of the people that lived in that village had grown a special type of mushroom and worked out a way to process it. And they invented what is called Quorn meat. So Quorn, Quorn, is a meat substitute that's available in superstores. You can make sausages and burgers and stuff out of it. Mm-hmm. And it was all came from this humble mushroom. And so they released this to the market, and they said, anyone can get this. It's the cheaper, healthier alternative to meat. And you know what? It flopped. Nobody wanted the cheaper, healthy alternative. That was the major thing that they were promoting when they first did it, was its health component. Was its price component. So they ended up removing it from the market because they couldn't keep it there. A few years later, they returned, and in this time they doubled its price, and they emphasized its health basis. And now Quorn is fully established as a really good meat substitute. It's just, there's a lesson there about how to bring ideas to the broader publics, because trying to shift to a more plant-based diet is incredibly important, and it's a hard thing to do, and it's little things like this that are going to make all the difference. So I think it's excellent, and I love the way it's reflecting in the students' work in the posters. I think that's brilliant to see. Nicely done.
And so as we look, I guess we're going to see some of those recipes popping up here in the next week. We've had some of the different legumes. That was the third episode from Beth. So thank you.
And Ted, if I can jump in here too with meat substitutes, there are some researchers from the University of Central Florida who were proposing eating insects on Mars.
Oh, there you go.
And can we get protein by eating bugs? And I posed to my fellow graduate students the would you rather question: eat vegan and have legumes for your protein, or would you rather eat insects? And it was like 9-1 in going with the legumes. But evidently cricket protein powder can be used to make substitute burgers. I'll just leave that there.
Yeah.
And there's a lot of cultures that they eat the insects. One of them is Mexico. Crickets over there are very popular, especially when they fry them and they put chile powder. So I've tasted them and they're delicious. They have even secret recipes with sauces and stuff. There's another one that is like an ant. It's bigger. I haven't tasted that. I don't know how they do it. Also in some beverages, which is the mezcal, which is similar to the tequila, they have a little worm on it. There's another. I have a podcast, and in the podcast they were mentioning different dishes with that. And one of them is it's like a wasp nest. I think they cook it somehow and they eat it. I don't know. I haven't tried that yet, but that's popular and it's not that gross. I've tasted just the crickets.
Well, I've had chocolate covered crickets before. I was in Japan, just off the Nakasendo Highway, and that was just quite a tasty treat at the end. I knew it was eating crickets, so I was very apprehensive. Crunchy. Oh, the chocolate, I think, is what got me. I ended up eating half the bowl.
They taste like grass. It's not something different. They are not like gooey or something like that, if they are fried, it tastes like
they're
grass.
crunchy. Yeah, for sure. Yeah, what we can do
I'll
is
stick with crunchy legumes.
I'll send you some, Lori.
I'll take the crunchy insects. I'm good with that one. Especially when you think about the Aboriginal histories, then and all of the background of the different things that they eat, worms, this in Australia, tarantulas, things like that. I think that part of this is just the word is associated with what we've learned culturally for it to associate with. But if I had to just stick with just legume, knowing like I'm on a nutrition plan right now, and believe me, there's minus the meat all the time. And I would just drive right over to Javier and say, bring on the crickets because bring them on.
Oh, that's funny.
And make them taste like bacon. Okay, but do that for me. But certainly we have to learn how to eat different things. We have to learn how to eat vegan. We have to learn how to eat insects. We have to change how we're doing this. And this is part of this whole discussion that we have all the time with Magnitude and the Exo Lab. But the more diverse we become in our thinking and acceptance of eating different things, it's our survival at stake. So bring on the insects.
There you go. Well, I guess without getting too far afield with the insect thinking, if we were going to take insects to another planet, let's say going to Mars, they got to eat something. So it comes back to, that means you got to grow plants.
Yeah.
Right. And so,
but could the insects, if we turn them into powders and things like that, could they be helpful? So I'm seeing somebody's saying the owner. Who's the owner? Because there's somebody waiting with their hand up and they're shaking their head to this. I'm wondering if they have more science to this than I do.
Oh, yes.
Hi, I'm Miss Hawker. I'm not the owner, but I just wanted to say hello and chime in a little bit because I love the conversation that just happened. I used to be in South Korea as a teacher, and it took forever for me to try what they eat as a snack on the streets all the time called bundagi, and it was the silkworm. And I finally did, and it was great. And it was all in my mind that was the block. And I love that. We need to open up, like you just said, we need to be open and try new things, but that's tough sometimes. So this is a great conversation to lead in another direction because we're going to the next frontier and we need to know, hey, just be open and it's okay. So thank you.
So for Exolab 9, should we start looking at some bugs?
At least as compost, something we can put in the compost or something in the medium. It would be really interesting to see what it does.
Mezcal or tequila could be the medium.
We'd have to card all our students then. That wouldn't be.
Chocolate covered medium.
There you go. So what I had in the program here, we can kind of keep it open-ended. But what I wanted to do is kind of recognize how, Tony and the team at Magnitude and I, how we kind of came up with the Magnitude Live was because of this pandemic, right? And we were scrambling to think about what the last part of the spring semester might be like. How is it as a student, if you're listening? Has it been tough not seeing your friends, or what's it like learning when your teacher's on a screen? That type of thing. And so I wanted to kind of just have an open conversation, whether you're a university student and you're just trying to do your finals, how has that been different? I think it's probably all of us are going to need to think about what school's going to look like in a few months, whether your school district's starting early, whether they're starting late, whether you're going to be able to go into a classroom. Will you have all your friends there in the classroom with you? Will your classroom be half the size, 1/4 the size because of distance, social distancing? And what might that mean from a learning perspective? What tools have we found are beneficial to help us out? One thing that I think was built into our DNA as a company at Magnitude is to be adaptive and to think about how we can bring learning right to the very edge of discovery in the simplest way as possible. And so I'd just love to hear from you, kind of an open discussion. What have been some frustrations? What do you have a thing that you love now that you never used before? And that can be from a student perspective. If you're on YouTube, I'll check that out. Our teachers here, love to hear from you and just have that conversation for 30 minutes or so. Or if we want to just kind of talk more about our legume experiment, their questions. Going to keep it more open forum, and I want to do more listening than talking. I usually talk too much. So what have you guys experienced? What's been frustrating? What have you found an interesting solution? What are you looking forward to? What do you still think are challenges? It's really kind of an open floor here.
Well, in kindergarten, it's unusual to do blended learning with them because they are one-on-one in my school district. But haven't heard from anyone trying to mix the technology with Google Classroom and stuff. So I've been experimenting with that for over 2 years of how kids learn with the technology in blended learning environments, meaning that I have class with them. That class is explaining a little bit about what they're gonna do. For example, in a worksheet, they have an activity there. I'm explaining, for example, they have pictures and they have to write what the pictures are. And then at the end, we get together, we discuss, and that's how we do these kind of activities. So whenever we got the pandemic, my students already knew how to do that, but my the other teachers in the district, they didn't. So what we created was a framework for the other teachers to piggyback in our platform. And we have a lot of challenges there because my students were very advanced with technology, with blended learning. And for the other ones, it was something new. And we have a lot of problems also because we didn't have our devices at home. And they have to be, the learning design has to be centered towards cell phones. So what I did was taking away the Google Classroom and start from scratch on the learning curve. So what I did with a team of teachers was to create a set of slides for the students to have like a roadmap. Like it was a video game, it was game-based learning as well. So they have several pit stops, like in a circuit of a Formula One competition. So the first step or first stop was language arts. The next one was math. The next one was social studies and science. So they have to go around completing their activities. They have 5 or 6 activities in each pit stop. So that experience with that and also the platform, we have 2 versions. One of them was in Google Slides, so the parents that still are able to manage the logs that we have in our systems, they click on the links and they have access to the Google Drive. For the parents that didn't have access to the Google Drive and still have a lot of problems, we save all those slides and we put them on a PDF. So that way we have 2 versions, one that is more interactive and the other one was more static and didn't consume a lot of more data. We just downloading the document and the teachers were, or sorry, the parents and the kids were working with the cell phone. So it was pretty interesting. We have a lot of engagement from the parents, from the kids. The kids were skyrocketing the activities, and I was saving their works because one of the challenges also was how are they showing growth. So the parents, what they were doing were taking pictures and sending those to me in something that it's called Class Dojo. So I have all the pictures safe in there, and whenever I have to go back, I just take them and I can see their pictures. So it was pretty interesting to see how they developed this, and I have a couple of kids that send me over 100 pictures.
Wow.
For 4 weeks of last day of school. So we have a lot of engagement, and one of them was the Leguminaut challenge as well. So they were doing their activities besides what they have to do with reading, writing, and math. So they were doing both at the same time. Sometimes we merge some activities with writing, and we embedded those in the lesson. So that's how we kind of tackle the challenge.
That's fantastic. It's different than what you would normally have in your classroom. Were you able to get 100% participation with your students?
Well, even in the classroom, it's not 100%.
So they might be sitting in the seat, right? But are they paying attention?
Yeah, but we have a lot of engagement. You know, it's usually the ones that don't participate too much, or there's not a lot of family contact with the teacher. They kind of struggle, but the majority of the students were able to participate.
How's everyone else been handling that? Whether you're a student or an educator, these 8-10 weeks that we've experienced this, we'll more than likely experience if we have summer programs we're doing, or definitely and probably see it in the fall as well. So how have you been able to manage that? What's your go-to resource, or what are some challenges you're still facing?
So we already have a homeschool. Sorry, it says, hope I don't lose you, because it just told me that the battery's down to 10%. I don't know why I've been plugged in. Hold on, let's do it again. Sorry about that. I hope I don't lose you. So I'm going to talk really fast. Yeah, so we have a homeschool program. I opened up under my own program called Science Yourself, and it's nicknamed the Space School now. The trickiest part of this is now getting everything packaged. Now, Michael was really helpful with that, because he created a system where he packaged his lessons and some of the materials and things, and then they could come and pick it up somewhere. Of course, it has to be sterilized and all of that before you even package it, and hand glove it to where it has to go. So now it's about getting a pickup point, or having somebody come to your home to get it, or you have to deliver it. So that would be me. But in the long run, there's only right now 8 students that are involved. So it's not that many children that we have to deal with. So in the long run, it's paying off, because they're able to get the materials. It's also sometimes just something I can send online. That makes it easier. If they already have these materials in the house, they indicate that to me. There's no pickup involved at all. In terms of teachers, the toughest part with teachers over here in Canada is helping them bring it down to earth, because they hear the word space, they hear the word International Space Station, and immediately they think, oh, that's too big, right? So trying to frame this in a way that it's about the plants, it's about making our planet better, it's about us sustaining ourselves, is actually getting through now. When I just started talking directly about ExoLab, they're like, not even realizing the benefits yet, because it was out of their scope and field of knowledge. But now it's starting to seep into some. We've only, as of yesterday, declared that our schools are closed, because we were initially going to be opening them as of next week. So now we have a better opportunity to dialogue with teachers and see where they want to go. The other thing is that in Quebec, we are primarily French, but we are bilingual province. So we have the other additional problem of, at the elementary level, all science is done in French. Can you imagine talking about the things we talk about right now, even today, in French to a child in grade 2? It just will go over their head. They're still trying to learn le soleil, the sun. They're still trying to learn les planètes, the planets. So I'm looking for ways and means to help them. The drawings, the illustrations kids do, videos, this program, the website, the Facebook page. These are all the links that I'm trying to feed them, so that they get a little more comfortable in thinking about getting on board with this. It is a down-to-earth program, always in my mind. But at the same time, it is a program that's going to benefit so many children. So my goal this summer, we've already started our planning for our space school. We will be online, and we're going to be bringing a good chunk of this into the kids and into their homes. And we're going to just keep going. I always think that no matter how many seeds you plant, somebody's going to pick up from that. And we're going to plant seeds to the keeners. And the last thing is just the differentiation. So Javier, you hit on that. In the schools and in my space school program, we're used to doing all of the multiple intelligences, and the movement is a key part of it. So we're looking at how are we going to bring that to the screen. So when we're zooming for the younger ones, we're going to actually have them grow like plants. We're going to do that with them. We're going to put on plant songs so we can bring the music to it. So we're right now 24/7 building this program to get it going. And then we will invite teachers in to see what we're doing so that they get it, that it's much bigger than just the idea of putting a plant in space. It's about the total package of learning. And lucky if it goes to space. How cool is that?
All right. Well, that's
as Diana was saying, you hit all these different subject areas. You hit English, you hit art, you hit math, science, even history aspects. So it's a great way to work cross-curricular in a project.
Absolutely. Yep.
There's a interesting, you look at how some areas have tackled this issue in the spring, and to see what happens in the fall. LA Unified School District, for example, work with their local PBS station, and they broadcast curriculum, right? If you got 97 or 98% of households that have a television, well, that's pretty straightforward. The families that are impacted, they may not have an internet access or a computer if their district isn't a one-to-one district, which is every student gets a computer. There are many places around the world where that just isn't happening right now. I think it's moving in that direction. But in the United States alone, I think 1 out of 4 households does not have a broadband access. So probably have mobile phone. It might be the parents, but do the children have that, et cetera. So this is interesting, using the free and reduced lunch pickups, these programs that in the United States work now. If you're a family in need, breakfast and lunch is usually provided for your kids. They keep those programs running, and that's a wonderful pickup and drop off point for material for these experiments. And so working with there in Lodi, I think that's a program that we'd like to be able to implement, getting things like seeds in the hands of the teachers being distributed through the free and reduced lunch programs, if that is what we're dealing with as we get into the fall. But, we have to kind of rethink. Can't just expect to take the 30 some odd students we have in a classroom environment in which we can directly have a dialogue with them. And now we've got this diversion of a lot of talking heads and kids being distracted, all different ages. How do they stay focused and engaged in what's interesting to them? So we're very curious in this, and it's kind of in a way, our meta experiment to thinking about other ways we can introduce tools to help us in our mission to nodulate a legume with this Rhizobia bacteria in space. But it's fundamentally, pedagogically, how do we now deal with this in a remote environment? And for our students, if you are coming in early, wanting to talk about the balloon launch, we'll start that in about 20 minutes or so. I'm curious if you're here as a student, what are some frustrations you might have experienced, or what's boring you, or what's exciting, or what do you think next year might look like? And I'll check that. Any teachers want to weigh in also on some of your feelings or experiences? Or Sarah, as a university student, what do you think? How's this going to transform university life, right? It come the fall, must be different.
Yeah, and I'm looking at doing a TA thing later this summer, so I also have to worry about teaching on top of that. But I think the main problem with the classes that I've been taking is they're not quite colorful enough. Like, it's usually the teacher just lecturing and then the class is over. And I understand that that's not maybe what all of you teachers do, because you're used to trying to engage younger students. But I think the teachers are realizing that shorter, like essentially videos that they upload tend to have better engagement from the students, and then also more like activities that the students can do that add that color and again engage students more than just a video of someone talking would. And so this Leguminaut challenge is really great in doing that and engaging them to think deeper about things and actually get their hands on some plants.
Right. I think that's what we're trying to do here, figure out a way to make that happen. Any other experiences or concerns as we think about the fall coming up here, anywhere in the world that you might be? Go ahead, Lori.
Yeah, I can add for the fall, the challenge for sciences will be the hands-on aspect, will be the labs. And that's something that in the feedback I received from my students is that that's the part that they really enjoy, that they find fun, and that they learn so much for that when they put their hands on it. And so if we're going to be doing online learning in the fall, that's going to be the challenge for teachers. How do we get them hands-on pieces, mailing them seeds? You know, that's what I've done with some of my classes. It was a little labor intensive getting 75 packages out, but you know, you have to go to great lengths. And so, but for a variety of experiments, how do we get them the supplies that they need to do individually to have that hands-on learning? Earth Day was a great way to get them outdoors. So we did a scavenger hunt. So you have to be creative. If we do online learning in the fall, it just cannot be all computer-based. We have to mix it up.
Right. The classroom is the world around you, right? In a way. And or the instruments that you need, you'll find in your kitchen cabinets, right? Or down in the garage somewhere. And so we need to be thinking along those lines.
I'm curious. Sorry, Ted, go ahead. I'm just curious, Lori, was there any discussion about opening up your labs so that only on certain days, certain students go into the labs? That's what we're planning with our universities here in Canada.
Well, for the spring, wrapping up this year, there were no students on campus.
No, I mean for the fall coming in.
Right. And I couldn't even take them things. I had to mail everything. So
Right.
for the coming year, we don't know what the picture looks like yet here in Florida. We are hoping that we will be able to start up in August in person. And the school is doing everything that's recommended to be able to have on-site classes, but we just don't know what the world's going to look like in 2 months. And they said for us to be prepared for plan A, B, and C, and then probably plan D, E, and F.
Oh my goodness.
Wow.
So,
And you see, that's the learning curve for us. But you said something earlier about creativity, and that's a big part of this. We're really learning how to be creative so we can be resilient in all of this. And we almost need to make note of this as we go, because this is really a game changer for all of us. And just quickly, because I'm going to have to go, folks. Tony, you had sent the ExoLab stickers and stuff ahead, and I did get them. So I wanted to let you know that as I asked you about the little boy across the street. So I have new students that were not my students. They're my neighbors, and they got involved. And so when the little boy came across the street the other day to pick up a new seed that had a little root on it, and I gave him the sticker, and I said, this came from a gentleman and a team of people who are out in California. And he was like, really? And he ran across the street, little plant in hand, little sticker. Dad! That's all I could hear. So I want you guys to hear that story.
Oh, wonderful. Thank you, Dana.
All right, I've got to go, folks.
That's okay. Good to
All
see
right,
you.
I'll be watching for the balloon launch though, Ted, okay?
Oh yeah, well, let's hope this one goes further than the Rockies this time.
Send it to me, come on.
Yeah, okay. This one looks like it's heading out to the Atlantic. We'll be talking about that in a couple minutes. One thing I will note with respect to labs, the California State University system is the largest university system, I think, in the country. 500,000 students, freshmen to senior. And that will all be distance learning in the fall. They've announced that yesterday. But what's interesting is the labs will be done on site, but they'll have to do it in breaking the classroom up. So I'll be really interested to see how they approach that and how they solve it. In my view, what I'm trying to do is imagine that we're going to have to make the labs work in everyone's kitchen. right? And if that's a requirement, then we solve it. And if we can get them back in the classroom, then that's even better. But we're trying to build everything right now and listen to where the pain points are and what we're doing specifically for Leguminaut Challenge and for ExoLab 8, but just in the general sense, right? You know, what is like the Edpuzzle tool that Lori shared with us a couple weeks ago, in which you take a video snippet and then annotate time and then ask questions around that video. I think are fabulous types of tools in thinking about how to solve some of these problems, kind of combining in a way, you know, what best practices are.
Hey,
so,
Laurie, FYI, I was able to embed the Edpuzzle into our classroom.
Wonderful. Yeah. Wonderful. I'm going to be working this week on taking more of those segments from our past live videos and turning them into Edpuzzle videos that I'll send to you and that we can share broadly.
Right.
I think authentic assessment.
Yep. I can put a number of lessons that is built with puzzles.
Mm-hmm. And as Sarah mentioned, the length of videos can make a difference. If they're way too long, 30 minutes, an hour, you lose student interest. And so we're going to need some really high impact, high content, intense videos. So maybe some of our PIs can help make some very short targeted presentations that we can add into those lessons. Because it's just a fabulous tool that is being used to get students engaged in the content and then to get feedback about what are they really understanding.
Well, I encourage all the teachers here active in Zoom in the program or interested in the program to think about what you might want to bring to your students. Maybe it's a short 3-minute video, or make a list of things you'd like to see. And collectively here, we can all kind of go down the checklist and help prepare that kind of content and information. One thing that Shivar, I don't know if you're still actively logged in, but I'd love oh, fantastic, to give us some feedback of your experiences there in South Africa. Now, you've got an informal learning program. It's really beautiful what you guys set up there at iInnovate. So I want to hear a little bit about that and maybe what you're doing with respect to the distance learning. And I have to thank you for going through and being really meticulous about cataloging timestamps of all of the videos of these sessions that we've done. It'll be a starting point for teachers like Lori and us here to say, that's a segment I need to go back in on and take those 3 minutes out that are essential to where we want to take our investigations. And so we've got this corpus of work, this body of work that spans now 50 some odd hours. And thank you for cataloging at least a rough draft overview. Anyone that wants to take that, we probably need to publish that, Tony, get it out there and people can do it and annotate on their own. They can use something like Edpuzzle or another tool. They can just take snippets out. But let's start taking this really large body of work. A lot of it, I have to thank Dr. Barker. Thank you, Richard, for really providing a lot of the meat, if you will, a lot of the vegetable protein that we need to make this substantive across all grade bands. I think you've even captured a few university students in our orbit with your knowledge. So thank you so much for joining us on our escapade.
Yeah, Laurie, to answer your question, I will put it on a web page with all the time codes and everything. Who's what topic they are talking about. I'll try to tackle that this weekend.
Because this is one of the really fun things about using astrobiology to teach biology in general. Because there are these moments when we're talking about these things when you really do need the rocket picture appearing and taking off, or the space station, like there. So as we've gone through these slides, we've talked about these stories, but like it's this secondary level of editing that just kicks the whole thing up a notch. I love the Edpuzzle component to it. Those questions like showing the students going through the sections. Hopefully those questions being relevant to like making them remember stuff. But a little bit of effort on that video editing, just the rocket taking off here and there.
We like that, huh?
I'll try.
did we lose Lori?
Frozen.
Yeah.
Frozen again.
She'll pop back. Well, Shivar.
Yeah, but let me take it away. What I want to say, well, the reason I timestamped everything was because all the back and forth discussions that have been happening here, those feel like proprietary bits of information. Like there's a lot of things you can find here that you simply just can't go on the internet and find. So I asked myself, how could I forward all the information here and all the concepts discussed to the educators in the schools in Cape Town, such as Fatima. So that's what I was doing. And every time a concept was discussed, such as, planetary ecosynthesis, terraforming Mars, gravitropism, all of those things, I'd timestamp it and I'd forward it to her, and we'd engage with students who could talk about all the concepts discussed and applied to ExoLab. Mm-hmm. And there's a lot of challenges we face here in South Africa, 80% of students don't have access to data, to internet and things like that. So we have to do a lot of things. We have to make sure they have small data packages, even if they can just communicate on phones, on WhatsApp groups. We have to see how we can constantly communicate to them, even if it means phoning them. So we do all these things and we bring those concepts back and forth to them. And it's quite amazing how many students we've had engaged. I think we've had about 49 students engaged, and we've managed to bring in over 100 hours of lessons right now with ExoLab. So it's been quite good. I know Fatima actually wanted to come on this call today, but I think she got caught up in something. But I'll see if I can get her next week and she can update you on everything we've done with ExoLab. But we have managed to overcome a lot of challenges, and it's been quite cool.
Well, let's do this. If it still fits the school schedule and your calendar year, let's get a South African episode running that's time appropriate for you guys.
That would be fantastic.
Yeah, and get some students on. So suggest to us what some good possible dates are, and let's get that on the calendar in the next couple of weeks. I'd love to do that. And you're UTC essentially 0, so we can bring in our European friends and Kenyan students and whoever else wants to join in. That's a little more appropriate time zone for you guys. So let's make that a reality. I'd love to do that and to hear firsthand. I know when we have a chance to chat with your students, it's always enlightening. And how do you solve that problem with the 80% of the students don't have access to data? There's an interesting project in I think it came out of San Diego. It's essentially internet in your pocket. They use a Raspberry Pi, and they basically take the equivalent of like a Wikipedia subset, and they load it all onto the data card for the Raspberry Pi. And then they distribute it to the students. They don't have internet access, but they have a $35 computer with all the knowledge that they need, videos and other information. And it's asynchronous, but it's kind of like pulling out the old Encyclopedia Britannica, right? And getting your knowledge there. It may not be the freshest thing since this morning, but a lot of stuff doesn't need to be the latest and greatest. It just needs the foundational information. And so that's an interesting way to kind of solve distance learning, perhaps. And I'd like to find out more from that company. And I think at Magnitude, we're also thinking about what we can do. Can you imagine if ExoLab actually had a resource of knowledge already embedded in its system? And so even though you didn't have internet access, it was cataloging it, recording it, and locally you could reference that data. And when you had a chance for internet connection, it then uploaded all the relevant data. That'd be kind of cool. And I'm way out of class here, so Tony's ready to kill me. But it's like these kind of ideas pop up in your head when you start thinking about how to solve a problem, right? I can't say that would be ready for fall, but these are the types of things that get us excited about really making a difference and providing the tools for the student in the most practical and meaningful way. Because, how do we get rid of the friction, right? And if you're excited about something, if you're curious about something, let's get out of your way, give you that information, kind of get out of the way so you can explore on your own. So any other thoughts about the future we face here in the kind of shelter in place or this social distancing and how it might affect school, whether you're a student or a teacher in the coming months? I think it's going to be an ongoing topic for us. No? All right. Well, I'm going to segue from...
Ted, I will add one more thing, and that is, in so many aspects, when we're in traditional lesson planning as teachers, that we're moving through so many topics so fast. And I think that this situation is going to force us to slow down. and to really prioritize the most important topics and to become experts in something rather than to be a novice in so many little things. And I really think it opens the door for project-based learning because it allows for cross-curricular. And I think that if we are going to do more online learning in the fall, that teachers will have to work across departments and across subjects. But it can be phenomenal when you.
Oh, Lord. We lost your audio.
I'm gonna have to second that. I'll support her in that notion, in the sense that interdisciplinary project-based learning is like the new hot topic in higher ed. The concept that historically we've all been like isolated in different silos is constant through all the different academic systems. We have become an engineer, or we become a biologist, or we become an English literature writer. And that's so last century. That's so old fashioned. It's not the modern here and now. It's not the future. And so, but you know, humanity, we're reactive. We're not proactive. We've suddenly realized that this is the case, but it's going to take a long time before the cogs of our institutions, the academic systems, the teachers have to implement because of the old fashioned exam process. It's going to be a while before that sort of stuff catches up with the modern reality. And so, yeah, and so whenever you start talking about astrobiology, we start talking about putting legumes in space-like environments, instantly you've created a project which requires rocket scientists, requires biologists, it requires computer scientists, it requires all these different skilled individuals to come together to make the whole thing work. But in order for the student, the researcher to interpret the data, they now also have to understand all those things. So suddenly no longer is it like, oh, I'm just going to be a biologist because I want to avoid the maths. Now suddenly people to realize they've got to do a little bit of each. And the rocket science makes the whole thing awesome.
Yeah, yes, to append that, building a team is really where it is, right? That you're an expert in astrobotany and you have deep knowledge in these other areas as well, much deeper than mine. Know a little bit about a bunch of little things, but I'm not an expert in anything, I guess. Jack of all trades, master of none. But I love getting people together and hearing that and finding those and connecting those points. And if we can get the history teacher together with the language arts teacher, with the physics teacher and the chemistry teacher, and then boom, what you have this thematic that you can follow along. You can look through all the lenses of history or of science and have a broader sense of the world and say, you know, I'm not that good in that field of study. My friend is. I'm going to bring them onto my team because we can do this thing together. I'm really excited about the chemistry, about the physics behind what's happening. And I'm interested in engineering. I want to build something around this thing we're talking about. And that's how teams are formed. And that's how really we thrive and how the world, I think, extends itself. We need to have the specialists. You need to be passionate about your thing, but you have to be open to be willing to hear different perspectives. And sometimes a breakthrough will happen in this, almost feels like a tangential field, because they're looking at things, you know, orthogonal to what you normally would look at it. And that's where magic can happen. So it's very exciting.
And that's the piece we'll have to get students to figure out, is how to work in a team if they're online learning, right? If they're not around a lab table working as a group, that they will need to still collaborate with others. And I think that was almost a little bit of a missing piece too, as we were doing crisis management in the spring.
Right.
But it's so much opportunity with that in the fall.
Right.
No, I wanted to ask everyone. Last week, someone talked about I want to get certified to teach online. What do they actually teach to be certified? Does anyone know?
What is that? Didn't get the question.
What do they actually teach to be certified to teach online?
Oh, I got my master in that. My master's degree is in instructional design and technology. So according to the program that I was working on, was the pedagogy of learning science, how people learn. And one of the best ways was with project-based learning, because it helps you to create artifacts about a certain topic. So PBL is very important. Also, we had to do some training in how to create an online course from the beginning to the end. And we use a program that is called Adobe Captivate, which is kind of similar to what Lori is using. What is the name, Lori's?
Edpuzzle.
Edpuzzle, yeah, something like that, but it's more advanced. So for you to create an online course, you have to create a wireframe, which is like a PowerPoint. You have to be able to be proficient in by videography, which is taking videos, taking photos and stuff. We have to be certified also in music, how music affects people, how images affect people. You have a lot of background, a lot of things all over the place, so it has to be simple. It's more like Ted in his field, which is more like art and graphic design. Another important thing was voiceover, because sometimes we don't realize the importance of voiceover in movies and that kind of stuff. Voiceover, what else? Videos, wireframes for the videos. So it has a lot of content to be like a professional, but that's like a master's degree program to be able to be mastering all of that.
That was great. Thank you.
Mm-hmm.
And that's
Well, and Tony's certification varies state by state.
Mm-hmm.
Even for virtual schools, online courses, it depends. Anyone can offer an online course, but you may not receive credit for it. You have to demonstrate that the laws vary a little bit, even with homeschool programs. So
also, LMSs are very important. One of the most popular one is Google Classroom. So you have to be able to know how to navigate, how to grade, how to design stuff for that.
Yeah, I'm glad you guys brought that up. If we can get this together, we'll have a guest here in the next 2 weeks from Virtual High School. And their program's been around for quite some time. You think about those rural communities that don't have a physics teacher or a life sciences teacher that's accredited for like AP. They enable small districts to have an expert remote. And so that's their specialty. So hopefully we'll get Kim or someone from her team from Virtual High School to join us. They actually have a program that's connected to the International Space Station that, if I'm not mistaken, is actually open now for free. At least it was during the spring. So we'll find out more from her, and I'm hoping they can join us in the next couple of weeks. What I'd like to do is let's keep this kind of conversation going in the background as we explore finding the right legume to send to space or to launch a balloon around the world is what we're going to try to do. About this conversation about how we can adapt and change is something that's very fundamental to solving our problems. It looks like I've got a lot of my teachers now on that are working with us on the balloons from Lodi. See Diana, Sam, Mitch is here, Tate, wonderful. Ms. Hockley. So this is fantastic. We're going to move over and talk about our high altitude balloons and what we've been doing. Magnitude, of course, we have our platform for learning, a learning management system that's based around project-based experiences, tuned into the next generation science standards. So you have your disciplinary core ideas, what you're going to find in a textbook. You've got your cross-cutting concepts, these ideas that can be worked across any domain of the sciences. Like the scientific method or something like this, understanding these cross-cutting concepts. And then you have the project-based experience that requires science and engineering practice to come together. So that was essentially the DNA that we saw back in 2013 that made Tony and me say, let's start Magnitude. My kids were just graduating from high school, getting into college. and Tony's daughters were just getting into high school. So it was like we saw this transformative moment. So this is really important and exciting to us. So let's keep this dialogue going. But now let's talk about sending something into the atmosphere. It's not going to get into orbit. So we're going to be really reliant on the weather. You know, what's the weather like today, and what's it going to look like when we launch our balloon, type of thing. How does a balloon actually float, right? You can use hot air, I guess. You know, hot air rises, as they say, thanks to gravity on the space station. It won't, which is kind of interesting. You have to ask yourself that question: why won't hot air rise on the space station? And then, or a gas lighter than the atmospheric concentration of gas that we have on our planet, mostly nitrogen. 3/4 of it's nitrogen and some oxygen. You got these trace gases like argon and what have you, but most of it's, like, say, nitrogen and oxygen. So we have some lighter gases like helium. The molecules are smaller and they're lighter, or hydrogen. So we're going to be relying on helium gas to lift our balloons up. And the first thing we want to do is understand what that actually is, this idea of buoyancy. And you guys can experiment with this at home. I'm assuming a lot of our students now are joining our teachers that are here in Zoom. They're joining us via YouTube. And if you've got your own communications channels on, just give me a shout out if they've got a question or what have you. And I also want to just give a big welcome to Liam. I see he's joined us just in time for the HAB launch. And Liam, it will help me quite a bit as we get to the launch part of this. And he's picked up a lot of the knowledge as well. He's getting experience just like we are about launching these balloons. But what I want to do in the 30 minutes here up to our launch is introduce to our students and to our teachers what we're doing and how we're doing it in preparation for our first attempt at a circumnavigation. So next Friday, about the same time, 11:30 Pacific time. So essentially 1 week from today, we're going to go into the same program, but we're going to try a little different balloon. And we're going to see if we can't send it around the world. So how's that even going to be possible, right? So let's start with our instrument that we have now. I took the liberty of preparing the balloon for flight last night. It's basically just a Mylar party balloon. I don't know if you guys can see that there. It's just, it's a larger one than you'd normally see, like Happy Birthday at the grocery store or whatever. This thing's about 1 meter across. And if it's kind of hard to see, pull back a little bit, you can see I was kind of being cheap. I didn't fill it up a lot. And so it's mostly empty. And the reason being is pressure changes with altitude on the atmosphere. So if we're here on the surface of the Earth, we've got more pressure here than when we go up in space. And if you imagine that this is our balloon right here, and we're here on Earth, we've got a lot of pressure from the air pushing on our balloon. It's going to look like it looks right there, which is a little bit of gas in there, and it looks mostly empty. But when it starts going up in the atmosphere, and so the balloon is going to want to fill the empty space and starts getting bigger and bigger. It gets to the point where it's all filled up and nice and large. And if we have too much gas in there, it's like blowing bubble gum. It's going to burst and that's it. End of our experiment. And we're really grateful for a NASA scientist, Bill Brown, who's done the calculations. He writes everything down. Each flight has got measured down to the 1/100th of a gram in terms of our weight and requirements for lift. But you can try this at home if you just get like a little tennis ball or your little rubber ducky in the bathtub, or fill a bucket of water. You put something down at the bottom of the water that has air in it, like your rubber ducky or like a tennis ball, ping pong ball. You let it go, what happens? Right? Pops up to the top. So if you can visualize this is really easy for me because I'd like to do this when I was a kid, and they don't let me do it now because I hurt people. But have you ever done a dog pile during the summer or the spring? It's a great day and you're having fun with your friends, and someone jumps on somebody else, and suddenly everyone's jumping on everyone else, and you've got this big pile of friends laughing and someone groaning because they're on the bottom. Imagine instead of you and your buddies, your molecules. You might be a nitrogen molecule and your buddy's there, an oxygen molecule. And the ones at the bottom of the dog pile are like they're here at the surface of the Earth. And your friends that are on top of you are the other molecules. And as you get near the top of the dog pile, there are less molecules, there are less friends. And of course, if you're the friend at the top of the pile laughing because everyone's underneath you, you're like that little molecule that's up in the stratosphere, way up in the atmosphere. And there's a lot less molecules pressing down on you. That's why we have less pressure. So just imagine this big dog pile of molecules here on the surface of the Earth, where it's kind of much more pressure. Now, compared to some other planets, this is relatively a mild pressure. You get to the surface of Venus, wow, look that up. That'll take a car and like crush it into like nothing. It's super hot there too. Venus is not very a good place, but it's got a very dense atmosphere. And that's our neighbor. It's closer to the sun than we are. And on the other side, everyone talks about Mars. But Mars has a very thin atmosphere. We have to go up about 19 or 20 miles into the atmosphere here on Earth in order to get the same density of atmosphere that we have on Mars. It's very, very thin. In fact, it's almost impossible to fly an airplane or a helicopter on Mars, but NASA is going to try to send a helicopter to Mars here next year, and it's going to fly in that really thin atmosphere. So our balloon is designed to fill up, and as the atmosphere gets thinner, as it goes up in the Earth's atmosphere, it'll expand and get nice and full, and it'll look like that party balloon you see at the grocery store. And so that is buoyancy. And weather is going to be important, and where the wind's blowing. So let me share something with you here. Share my screen, and let's see what I've got going on here. I've got too much stuff going on. I better clean my desk up before I share my screen. 1 second. Oh my gosh, it's a mess. Too many things here. This is what I want to say. This I love. And for those of you that have been in our program before, you know how much I really like this. This is called earth.nullschool.net. I'll put that in chat real quick, or I can't because I'm, anyway, but if someone can do that, that'd be great. Here's a view, a simulation of what's happening on our Earth from satellite information and from other high altitude balloons that have provided data, from buoys in the ocean that are measuring ocean temperatures and what have you. This is a simulation of what the atmosphere of the Earth is doing at sea level over the last 4 hours or so. And you can see here up in the Pacific Northwest, looks like a little swirling action going on. Down the coast of California here, it's moving offshore. It's interesting, look at that. You can see this equatorial line happening right here, which the winds are going westerly here in the northern hemisphere. And in the southern hemisphere, it looks like it's kind of moving easterly as it gets up toward the top, this little band right across the middle. Very interesting. But what's fascinating, of course, is just as you change, the pressure changes with the increase in atmosphere, your wind behavior changes as well. So with this program, I can do a lot of different things. This is surface pressure. And as we go up, we get less pressure. So you watch our number going down. This number is measuring something called pascals. Actually, they're hectopascals. And here on Earth at sea level, it's about 101,000 pascals. Here they've just rounded it up to 1,000 hectopascals. Where our balloon's going to go today in about 20 minutes or so, maybe more like 30, is probably right around half the air pressure. And look at the difference. Right, much higher winds. That little swirl that we saw off the Pacific Northwest is much bigger. So you imagine, just think about it in three-dimensional space. There's a lot of wind going up as you get up around 25 or 30,000 feet. And you can see the wind blowing here where we're based here in Berkeley, California. We're going to be blowing almost due east, and we're kind of on our way. So that is one aspect to consider. Another is it going to be a good day to launch? And so if you're joining us today thinking about, I want you to be thinking about our mission next week, because we're going to do the big balloon launch. And we'll have a couple more launches in there just for fun as well. But what will our weather be like? And I think you're able to see that as well. So here is a forecast. It's a prediction. It's not 100% accurate. What it's going to look like over the next couple days. And here we are on Friday, and you can see the time here. It's kind of set the band. And what I love about this is Weather Underground, but there is so much information just on this one page. We can look at this red line. This is temperature. And you see how temperature changes Why does it go up and down like that? You might have a hypothesis. Talk to your teacher about this. But you can see that it looks like the prediction says it's going to be going up and down every day. We can also see these nice symbols up top where it looks like it's partially cloudy. It looks like we're going to get some rain on Sunday here in Berkeley. And it's going to get sunny here by next weekend. Oh, that's going to be a nice balloon launch next Friday, if it all stays according to the prediction. Did you know you could actually do your own weather predictions as well? If you have something that can measure air pressure, high pressure or low pressure is a good indicator of changing climate, changing weather for you. There are other things as well. There's something called dew point. This is the point in which you get condensation at a certain temperature. And when this meets it, you might see like water on the grass in the morning or on your plants. Well, how did that happen? It didn't rain last night. It's got cold enough where the water from the atmosphere condensated on the ground. And so you can see a dew point measurement here. And when they cross over is when you're going to see water in the ground in the morning. It looks like we might be getting that just in the early dawn on some of these days. If you're not from the Bay Area or from Northern California, do your own homework and look and see if you might have dew in the ground, and then go out and see if you see it in the morning. Other cool things on this graph here, this green bar down here shows the percent humidity, or how much moisture is in the air as a percent. And you can see it looks like it's pretty damp here. We're in the 80% range, 84%. It's staying pretty high. This is interesting. It also has this up and down cycle. I wonder why that is. You might want to do a little investigating. In addition to that, we've got pressure. So pressure isn't constant. I mentioned the 101,000 pascals at sea level. That's an average. It goes up and down depending on the seasons and on weather. So you can see we actually get a big drop in air pressure here predicted, but also look what happens. Well, does that correlate with rain? If the pressure falls, does that mean it's going to rain? I don't know. According to our prediction, it looks like by Sunday, you can see this blue graph right here. It looks like it goes almost to like a 83% chance it's going to rain on Sunday. So it's not a good day to go to the park. Probably not a good day to launch a balloon. But in addition to that, I'll just go all the way down to the bottom here and look through this on your own and investigate it, and definitely use a tool like this for our predictions next Friday, because I'm going to ask your teachers to have a couple of you, if you're interested, to share what you know. And we'd like to hear from you as you're learning about this, what you're picking up. But this is also showing us wind speed. And this is interesting. It kind of goes up and down also. I wonder why that is. But what I like here for this particular format is it shows us wind direction, or at least it's a prediction. Of course, it's never 100% accurate. But these weather folks, the meteorologists, they study this and they're pretty good about it. We can see the wind right now is blowing about 9 miles per hour, which is a little on the high side. I'm not seeing a lot of wind out my window here. And it looks like it's climbing. So by the time we get to what time is that saying? It's saying, oh, that's 5 p.m. Here at noon, it's looking like it's going to be about 9 miles an hour. And if I wait much later in the day, it's going to get really blowy, and I'm worried about it. We saw one of our balloon launches a few weeks ago. It pretty much ran across the ground and ran into the fence, and one of our neighbors picked it up and let it go. So hopefully it's not too windy for us on this launch, but you're going to need to make sure that the weather is good. And what's super important for us, of course, if it's raining, we're not going to launch. This is a very lightweight balloon, and the extra raindrops are going to hold us down, might even make us crash to the ground. And technically, if we use those super big balloons, I know in Lodi, some of you guys, I know our teachers have experienced it firsthand, but we launched the really big latex balloons, and the Federal Aviation Administration wants to know when we send those big balloons up. 5 pounds or a couple kilograms of mass that we're sending up. It's going up 19 or 20 miles. They want to make sure they can see it and the airplanes can see it. Not a lot of airplanes in the sky right now, but still we have to contact the FAA. This little guy, the little balloon with its payload that we're going to launch, we're not going to need to worry about that with the FAA, but we still want good weather. And so right now we're looking at today. Everything seems to correspond to a good day to launch. I'm looking out the window just for visual observation. It's looking like maybe I'm starting to see a little wind here, but outside of the heavier wind, I think we should be okay for a launch. So in addition to that, we're going to need to do prediction because we want to kind of find out where our balloon's going to go. Now I'm showing you this on the video and I'm teachers, but I'm also going to give you step-by-step instructions. And next week, I want you to go and run your own predictions. And with your, if we can do this, if the balloon actually can go all the way around the world, each time you check on your balloon, it's going to be in a new position somewhere in the world. And you're going to need to want to run a new estimate because the future changes and it's not always 100% predictable a week in the future, or 2 weeks in the future. And so we're going to use a tool from the National Oceanographic and Atmospheric Administration, NOAA, to do our calculations. And the first thing that I needed to do here, and I kind of pre-filled it in, I'll just walk us through it real quick, but you're going to need to know where we are, where we're located on the planet. And our planet it's basically a sphere. It's kind of a little larger around the center because it's rotating. So they call it an oblate spheroid, but think about it just like a basketball or not soccer ball, but more like a basketball. And you've got latitude from the equator to a pole, like the North Pole or South Pole. And we met in degrees. Oh, remember in your math, like 0 degrees, and this is 90 degrees, a right angle. And if this is about 45 degrees. If you look at where I am in Berkeley, California, we're at about 37.87 degrees north latitude. So something like that from the equator. Of course, if I was in the southern hemisphere, maybe down in in North Africa or something, it'd be my south, 37 degrees south. So that's our latitude. And then our longitude is based on an arbitrary line that was set at 0 point at the Greenwich meridian in Europe. And then you go west, you count in one direction, or you go east, you count in the other direction. And in this case, you go to 180 degrees one way and 180 degrees the other way. 180 degrees west, 180 degrees east, and of course, 360 degrees makes a circle, right? Or think about the circumference of the Earth. And so in this case, we're 122 degrees west from Europe. And that's where we are here in Berkeley, California. These extra digits here just are making us a little more accurate. We're actually at the Berkeley Marina, as that's where I'll be heading. And so I placed think about it like a calculator. I placed my latitude and longitude so it knows where I am on the planet. And I'm going to set this here for a prediction for 192 hours. I'm going to look in the next week what's going to happen. And I want 3-hour updates from all the satellites in space and from the NOAA Institute and other ground stations and buoys in the Earth and what have you. And I can just click next. And here we go. And this is the different prediction cycles. So I can predict a new, this is the most current one. You'll notice the time is in UTC. So it's not our time here in Berkeley. It's based on universal time. We're 7 hours behind universal time. So a noon here would be 5 a.m. in the morning. And you can see the date. So I'm going to pick the most recent prediction. Click next. And time-wise, again, the hours, this is going to be 1700 UTC, which is about noon our time. We've got our latitude and longitude. Here's our altitude. I've got this preset at about 8,000 meters, about 25,000 feet or so. And I'm going to select AMSL versus AGL. And what do those mean? AGL stands for above ground level. So if I was 8,000 meters or, whatever equivalent height you want to think about above the sea, that would be AGL. But if I was 8,000 meters above a mountaintop, that's also AGL. Well, so that's interesting. I really kind of changed my altitude. So we don't want to use this one. We're actually going to use AMSL, which is above mean or average sea level over the course of a year. And so as it's traveling around the Earth, whether I've got mountains or valleys, water or whatever, it's going to measure the average altitude based on the mean sea level. So we want that.
By the way, Ted, it's 1900 hours UTC.
Okay, what did I do there? Thank you, Liam. Oh, 12 plus 7 is 19, isn't it?
Yeah, they're currently 7 hours on UTC.
Right. Yeah, I just don't know how to add. Thank you.
No problem.
And let's see, what else do we want to do? We want to add a meteorological field. If we have rain that comes into our path, or we fly into the rain, it might not be beneficial. Not much we can do about it, but at least it's good to know from a prediction standpoint. I'm going to want to track the rainfall in terms of per hour, millimeters per hour. And that's it. I request a trajectory. It's going to do a little calculating. It's going to take a minute here. It's going to pop up where it thinks it's going to go if we launch here at noon. And I'll show you that. And then Liam, since you're here, I'd love if you've got a sky tracker with you, right?
Mm-hmm.
I'm wondering if you can walk through this device after I show this map, talk a little bit about it, and I can get on the road.
Sure. Let me just, I want to go grab it.
Yeah, okay. And I'm going to talk about this for another minute. So here's our, wow, that looks really weird today. This is what it says it's going to do. And this is different than what I pulled up last night, of course, because things change, right? And so this prediction happened 12 hours later than my last one. Looks like we're heading south down toward Arizona, getting down in like Sonora, Mexico. I think this is where we lost our last balloon. That one kept on heading kind of southeast. And then we're going to do some jogging. We're going to do this weird change and going northeast all the way up toward Canada. And we're working our way toward Diana.
Yeah.
Out there, Wisconsin. We're going to see right over Dr. Barker's place. We'll say wave to him. And that's going to be how many days from now? Let's see. This is a midnight UTC when you see the big triangle. So that's, and here we are at noon today. So here's the next day, midnight. So here's what it's going to look like noon tomorrow. Anyone better with their geography there in Arizona? Is it near Phoenix or something? And then it's really kind of taking its time down here. Not a lot of wind. It's just going to float around according to the prediction. And it's going to spend its third day just south of the United States. Fourth day, working to New Mexico. Fifth day, is that Nebraska? I'm kind of bad with my geography. Heading up the wind picks up here, it looks like. We're in Canada. 7 days, we're over the Great Lakes here, and we're working our way back down. Maybe we'll see Lori in Florida. So just over the course of the week, South Carolina, we're on the way and heading off the coast. Now, this is the smaller Mylar balloon. And so typically, if we can do it right, it goes at least this far. We've had some troubles the last few launches. So it'll be really great to see it go this far. But we've also had them go all the way to Africa. And we had one we launched from Kentucky here. And we lost it on the way to St. Petersburg in Bulgaria, which was really fun to, disappeared for a little while. We were relying on the radio, which Liam will share more about the radio here. So that's our predicted flight path. And you can see here's that latitude line. There's 30 degrees latitude. This is 45 degrees latitude. And here I am around 37, right? And here's your longitude lines, right? We said minus 120, here's 90, 105, 120. So 0, or the Greenwich Meridian line, is over this way over here. And if I look at the rainfall, ooh, that looks nice. Not much rain showing up in our prediction, pretty much 0. So that's nice. So that's a good overview. Just check, I'm going to stop sharing my screen. I would just check our, if there are any questions.
What's the call sign?
Oh, it's kilo
dash
6.
UQ dash
5. Yeah.
Dash 5.
And so I guess that's the last thing I'll share with you. You can pop it in. You're not going to see anything now, of course.
Yeah.
I'm going to suit up with my mask and get in my car, grab my balloon. It's going to be about 10 minutes. I'll join us via my phone. And I'll turn it over to the one and only Liam.
Over to me. All right. So you're heading out right now?
Yeah, I'm pretty much just getting my phone set up, and I'll walk out the door here.
Okay. Hi, everyone. Yeah, this is Liam here. You should be seeing me holding up this thing that looks like a miniature satellite. This is the Sky Tracker that was built, designed, programmed by a NASA engineer, a current NASA engineer, Bill Brown. who works at Huntsville, Alabama. He's actually on NASA's team that's building their SLS rocket that will take the Orion spacecraft into space. And in his spare time, this is what he built. And the key thing about this is, one, it is really very light, which means it doesn't require a lot of lift in order to go up. But what does it do beyond just looking like a cool satellite thing? It's a device that allows us to track the position of the balloon, its altitude, its latitude and longitude. It's also got a temperature sensor on here. So how does it do all of that? Well, on the one side, what we've got going on is it's got a GPS module on it. If I just bring it up there, that's the GPS module nearest to my fingers there, and there's a temperature sensor on the board there as well. Has anyone, some of your students may have heard of, and certainly the teachers may have heard of, a microcontroller platform. Basically, this is a version of a very customized microcontroller called Arduino. So if you've ever done anything with Arduinos, they're often used in robotics and other things. This is really what is on here. So how, what you need for any kind of microcontroller is power. Unless you've got power, it can't do anything. And that's the function of the solar panels. They're on here. Very, very tiny and very light, very thin solar panels. There's obviously two of them on here, and simple power wire circuit going onto the board. And that provides power whenever this has direct sunlight. And it really doesn't get enough juice if it's in a cloudy condition. It really has to be in very bright sunshine, which means that so when Ted goes out and launches this, I think it's perfectly clear today. But there's several aspects of this. So I mentioned the actual sensors it's got on it, but the other thing that we get from this is the actual position data. I'm wondering, is someone got their microphone on? I was hearing some squealing and stuff. Anyway, not sure if you're hearing me okay. But so the other thing, how do we get that information from this down to us? So on the actual device here, it has a radio transmitter. And you may notice that there are a whole load of wires coming from this. On this side, there's a wire. I'm sort of holding this side of it. And on this side over here, there's another wire. What that actually is the antenna. And it transmits all of the data, its location, altitude, temperature, and various other things, on an amateur system called the APRS. It's actually a packet radio service where amateurs all around the country have set up, and around the world have set up ground stations. They're really nothing more than a very specialized radio receiver, and in some cases a retransmitter, but mostly it's a receiving network. So when this is up in the sky, it will track all of that information will get sent down and then shared on a website. And I guess I can share my my screen here just to show you what that is. But essentially, that's what's going on. I'm just going to put that aside, and I will just share my screen. Here we go. So you should just be seeing the page I've got showing here is on aprs.fi, and currently it's configured for where I am. This is my it defaults to finding your location, and let me just pull that out of there so that I can still see everyone. All right, yeah. So, and as you can see, what it can do here is you've got to type in the call sign. You may have heard me asking Ted, what is the call sign for this tracker? And that's the key thing. So I mentioned it's an amateur radio program, an amateur radio system that is on here. And that's a system where anyone of any age, as long as you can pass an exam, you can get your own ham radio license. And those of us who are ham radio certified, what that means is that when that happens, you get your own personal call sign. Now, Ted's call sign is KK6UUQ. And the way that Ham Radio World speaks that, to make it clear so that you understand what it is, it's actually, we call it Kilo 6 Uniform Quebec. So that's just a way of making sure that you know that you're hearing the words correctly.
It just sounds kind of cool too, right?
It does, yeah. And especially if you're from Quebec, which sometimes we do have someone from Quebec, it can be very confusing. They're thinking, oh, why have you got Quebec in that call sign? But it really is just letters. My one is Kilo November 6, Echo Quebec Uniform. So KN6EQU. So what we've got to try and find is the call sign. So right on here where it says track call sign, and I know that may be a little bit difficult to see. Let me see if I can zoom in, just to make, yeah, it does make it a little bit bigger. Let's see. Anyway, so now you should see it's got a place for you to put the call sign in. Now, the one that is up that Ted has got, it has a -5 on the end of it. So programmed into this device is your call sign. That's done by Bill Brown before he ships them. So he programs
Liam,
them with,
pardon,
yeah.
can I interrupt you? I just saw my screen. Did I not unshare my screen? Are you sharing yours?
I'm sharing my screen.
Okay, mine's just frozen for some reason. It looks like I'm still sharing my screen. I'm sorry.
Okay, yeah. Hopefully people are seeing my screen. My cursor is going over it there. So I'm going to bring up a previous one, maybe -3, just so that will show. So here we go. And by default, we'll share a proper URL for the actual launch in a minute. But what I want to do is show this one. So this was a launch. Oh, this was the other one that lost itself over down in Mexico. You may have also seen I changed the track tail length to 6 days. That just means it'll show us the track for a 6-day period from the last time it was received. So I'm just going to zoom in down here. So, and this is approximately where the first transmission of that previous launch happened. It's cool seeing Ted driving there. I love seeing you on your way.
I'm looking for a place to park right now.
Yeah, I bet.
It's actually it's not too bad today. Here's one.
Very good. Very good. So, in just as I think, if I'm right, this is likely the place where Ted will be launching from. Is it the Cesar Chavez Park?
Yep.
Yeah. Okay. So, what was the one last week? Was that
same location?
Yeah. No. What was the call sign last week?
4.
Oh, that was 4. Okay.
Yeah. Yeah.
Of course,
it's 5
that
today.
makes sense. All right. So let me just change it to 4.
4
just
was
for the last
sad.
one. Although, you know, it sort of brings up, you know, sad memories in a way. But this one will.
Everyone's an adventure.
Yeah, they are. That's right. It's good. And it's also just recognizing the whole aspect of this, that these are experiments that we're doing here. So this is showing you after, so Ted launched it last week over here, and the very first data downlink that we got from it was on this one. So, and that was when it was at 900 feet. So it might be a cool thing to for us to decide how quickly we will see it on the new one after Ted launches it. So wow, there you go, Ted. You're sort of ready to go there.
Probably noisy too, huh?
Yeah. No. It's perfectly fine.
It's another beautiful day, that's for sure.
Yeah. So the other thing that you can do is if you have so this is my handheld radio that I'm holding up here. You look at my video. This allows you to pick up the radio signal direct. So in other words, this thing transmits, and if you have one of these, you can pick up the actual downlink of the telemetry, the data, using this, and it shows it on the screen. But what we have to wait for is for Ted to launch it, and then when it gets high enough and it's charged up enough, so although this has solar panels on it, it also has a tiny little battery. So I think it might even be more of a capacitor, but it essentially holds enough charge, and then at that point it will switch on and begin transmitting. It also has to get a satellite lock on the GPS system. You know, we're all used to that on our phones, but you know, when we switch it on, it has to actually receive the signals from all of the geostationary GPS satellites that are above the sky in order to triangulate its position. So that's why it can take a few minutes before we actually get the first transmission.
Liam, can you unshare your screen?
Yeah, I will unshare my screen. I'll stop the share. Yeah, so what I was talking about is you know, the GPS on here. So, and I think we're going to switch to Ted so that we can watch this. Oh, Ted, you're going to the top of the hill, aren't you?
Yeah. So there's Mount Tamalpais, Nevada. We're in. I got some kites here. I'm going to.
Oh, yeah, get away from the kites.
So there, I don't know if you can see it. There's San Francisco and the Golden Gate Bridge. But I'm going to launch. I'm going to untangle first here.
Oh, that's a beautiful angle.
Yeah.
Isn't that lovely?
Very artistic.
Yeah. I feel like we're all lying on the grass below Ted right now. I just love the
insect point of view.
Yeah.
The grasshopper.
A cricket. Don't eat me.
Very cool. I know I did a long monologue there around all of this stuff. Hopefully that was useful. I'm mostly just filling in the time for to let Ted do what he needs to do.
Is that a kite?
Oh, on the left there, yeah, it could. Or is that an insect crawling along the edge of Ted's phone? Yeah, that is. What is that?
Oh boy, so I'm all caught up here.
Take your time. Oh, that is just nuts, isn't it? What is that little thing on the left there?
Yeah, kite.
Yeah.
It's an octopus.
Yeah. Do you want a spotlight? Can you? I guess that doesn't matter. I guess we can spotlight it. Oh, you are going live. Do you want to spotlight Ted's video just so it keeps on him?
I have.
You have? Oh, okay. All right. Yeah. So this is just in the Zoom interface. It doesn't quite do the same thing for us all.
Oh, really?
Yeah, but you're right. I'll check the
Okay.
YouTube link.
Yeah, our antenna got caught up with the line.
Tony?
Yes.
Oh, there it goes. Make sure that on the Zoom, it's showing different faces. Try to see if it can lock on to Ted.
Yeah, I think what you'll find is, I'm going to open up the.
All right, there we go. All right. All righty. You guys good?
Yeah.
Yes.
All right. How about a countdown? We're going to let this baby go.
10, 9,
9,
8,
8,
7, 6,
6,
5,
5,
4,
3,
2, 1.
Is that a good shot?
That's pretty windy.
Yeah.
Yeah, I'm not sure it's spotlighted, Tony, just because it's switching between us all on the YouTube side.
That's because you keep on talking, Liam.
Yeah, I know. Well, it's yeah, but if you spotlight it, then it doesn't do that. Oh no, don't spotlight me.
There it goes. I'm going to guess in just 4 or 5 minutes, you're going to get the signal.
Yeah, I'm putting it in right now. KK6 UUQ-5 and still not found in the database. So I think it's we got to wait for that first.
That wasn't windy as the first one. That first one, I remember it was really kind of low to the ground. And I reweighted again this morning to make sure that we weren't getting a different lift, and it looked fine. All right, students. So what's nice here is that you can track this all weekend. And if we're arranging a time to chat with you and your teacher next week, we'd love to see what you think about where it's going to be. If you have your predictions, and maybe some other questions you can ask yourselves is, what's happening underneath the balloon where it is right now, right? What's life like? It's going over the Navajo Nation. What's happening over there? If it's going through Montana, what's it different, Canada or Mexico? If it actually gets over to a different part of the world, what might it be like underneath that balloon? Think about how big the world is, right? So many different things you can explore. How does it stay up there? If you were going to add an instrument, this one isn't taking pictures like some of those real big balloons we launch. What kind of instrument would you want to see on this? Maybe we can build something like that in the future. It's really up to your imagination. The balloon we're going to launch next Friday is going to be a different design. It's going to go higher and hopefully much farther. But there you go. I can still see that silver. I don't know if you can see it on my phone, but it's up there right now over, I guess that's over North Berkeley. So you should see it in another minute or two. All right. Do we see any comments or questions from the students in YouTube, Tony?
One moment.
And teachers there at Lodi or any other teachers that are doing this, following along with us, do you have any questions or ideas, thoughts?
Okay, some comments. Awesome, so cool, amazing.
I love that one. Like, duh, everything science is amazing. Yeah,
All right. Well, what makes it awesome?
We all agree that.
So help me understand when it's awesome, what's awesome to you, right? What specific level of awesomeness are you talking about?
Okay, Yodu is saying science is the fundamentals of life.
Yo Dude is Dr. Barker. What do you think? Anyway,
all right.
We're top of the hour. I guess we should probably sign off here. But we can track it. Did we provide a link in YouTube and on our Zoom?
Yeah, just trying to just waiting for it to have something there at the moment.
Okay.
Obviously,
I mean, I'll hang on. I don't need to hang up, but also
well, no, you don't have to hang on because you hanging on is not gonna make the difference with the signal.
I don't want to jinx it, right?
Oh, yeah. That's right. It's got that. Is Ted nearby? If he isn't, he doesn't care. And,
he doesn't care about me anymore.
So I'm not gonna
go in the wild.
There we go.
Well, I just realized I parked illegally. That's why I was a parking spot. Fortunately, no ticket.
Yeah, there you go. That's that would have been a nice addition to the.
It would have been $100. Yeah. According to the sign when I just made the U-turn. It's all good. Everyone's showing up. I guess it's a Friday noon. It's gonna have more of that next week as well. Maybe I should find a different spot to launch on next week.
Yeah, I know where we are. The beaches they've reduced a lot of parking, to help create more social distancing.
All right,
park.
so students, if you think it's awesome or just amazing, I would love you to write me a little bit more about what makes it so. And if you think about school, it's kind of weird, right? The way school is right now with like talking to your friends via Zoom or whatever, some Google Classroom, your teachers being far away. If school's like this again in a few months, what can we do that's totally awesome and cool, right? That's what we want to do. So give us some ideas, whether we're doing more balloons or doing plants in space. So your ideas help us. You're the fuel for our desire to do fun stuff. We'd like to say we're powered by curiosity. And so the more curious you get, the more power we get and the more cool things we can do. So even if it feels like an idea we haven't explored yet or something that's totally different, like you want to talk about things under the ocean, well, we're not experts, but we can probably find someone that is. And let's talk about that. The idea here, I think, with life as a whole is let's find the thing that really kind of gets us excited, gets us interested. Maybe you really like movies, or maybe you like music, or maybe you like writing or reading, or maybe you like science or engineering.
Hey, do you want me to chime in here? Sorry to interrupt. I thought it was key to know. We got the first ping.
Yay! What's our altitude?
1,000 feet. So
okay, good. It's not super high and it's not super low. So that's
no. So I'll post the link to. Does it allow me to?
You should be able to. And you just in comments on
the YouTube side. Yeah, for some reason it's wanting me to sign in, and I should be signed in. Anyhow, I'll if you want, I'll share my screen just to show what it's showing right now.
Awesome. Thank you.
And do let me know if you don't see my screen.
So, I guess one thing we didn't share for you guys meeting Liam for the first time, he's based in Southern California. And you saw he has a sky tracker just like the one I just launched. And so I don't know, Liam, are you up for doing that balloon race with us on June 1st?
Yeah, that's is that the one?
That's with Joanne.
Yeah. So that's the high altitude balloon.
No,
ones,
we're just
right?
going to use regular. We're just going to, what I just launched, we're going to do that. She decided to back away from the all around the world on June 1st.
Tony, I think you may still have Ted
yes.
spotlighted, so probably people are not going to see what I've just shared.
No one wants to watch Ted driving in the car.
All right, one moment.
cancel spotlight should be an option right there in your Zoom controls.
It's a beautiful day today.
There you go.
In my cave, I'm gonna have to get out in the garden.
So yeah, so we've had now the second ping. I call them pings. That's just so what we've got. This was the first one over here. Let me zoom out a little bit. So Ted launched it over here in Cesar Chavez Park. It headed pretty much a little bit northeast. And then the first ping here was at 1,000 feet. And then 2 minutes later, so every data point on this, it's 2 minutes between each data point. And the next one was 1,500 feet. So,
okay.
Yeah, so that went up a reasonable rate in 2 minutes. So that's 500 feet in 2 minutes between those. The other thing I just want to point out, this is just the nerdy factor of this. Zooming out to show you, wow, that when you hover over the individual points, can you see that there's a little purple line that's heading somewhere? That line, if I zoom out, oh gosh, it's so far away. That's pointing to the ground station that picked up that signal, because the ground station is literally in some amateur radio operators' home in many cases, just outside in their garden, or they have an antenna on their roof. And yeah, there you go. So this one, that was picked up all the way out in Santa Rosa.
Wow.
Yeah.
Oh, that might have been Tim Bosma.
Let me see what it says it is. And you see again, it very helpfully gives the call sign of the ham radio station that picked it up. That one is Kilo 6 Quebec X-ray. What's Y? My brain is Yankee. There you go. Kilo 6 Quebec X-ray Yankee -1. That was.
Oh, that's a newer call sign, so it's probably not Tim.
Anyhow, so that's it's on its way, and it's on its way up into the sky.
Liam, what happens if it rises too fast?
Well, if it rises
we're talking about the balloon.
Oh, I think we're having trouble hearing you, Ted. So maybe I'll try and answer that.
Ending, right?
Yeah. So you cut out, Ted.
And so it's about.
Yeah, you're still cutting out.
Phil, go.
Yeah. So
it's all you, Liam. Go ahead.
Okay. All right. Yeah. So the rate of it rising is purely a function of the amount of lift that's generated by the helium that's inside the balloon, and really the other factor is how heavy the actual payload part of it is. So it's the difference between those two. And with Bill Brown's very particular and very detailed way of measuring the lift, we should be able to maintain a pretty good an appropriate amount of lift for the payload that we're doing. So we shouldn't ever experience too much lift, but the lift is really determined by just the local conditions to some extent. But that's what determines how fast it goes up. So I don't think this is necessarily going up too fast. It could simply be last week's one. If I were to bring that up, what we were seeing was, and really going into the Null School website, where you see the ground level air versus higher altitude. So at different altitudes, you may find that I'll call them localized wind conditions may also play a factor in how fast the balloon rises. There may be other technical parts to that too. But that's really it. So interestingly, yeah, that really is happening right here, because we've now had another, I've been talking so long, we've had another 2 pings. So the second ping, it was at 1,500 feet, but the one after that, it was just below 1,500 feet. So it went from 1,578 to 1,489. So, and then the next one after that, it's gone up again, 1,700 feet. So I think that really does illustrate that certainly you may get a variation in the amount of altitude gained or lost between each ping. I think what we'll see is when it gets up to a higher altitude, we'll see that it pretty much keeps to that higher altitude. It's always a guess for us as to how high up we get. These particular balloons, if you have a very good filling of the helium and don't get a lot of regular air in it, it has been known to reach over 30,000 feet. So that will be a good check there.
Yeah, we're hoping for a 25 to 27. Something in that range would be ideal.
Oh, cool, Ted, you're back. All right. So
yeah, I ended my land. You got it all covered, Liam. You know what you're doing. It's exciting, though, isn't it, guys? So we're going to sign off here in just a minute, but I'll just do a quick round robin. Any of our teachers that are involved directly with this project, if you want to schedule some time with us, please do. Let's get you and or your students on a call. Any questions about the curriculum or what we can investigate? You should have all received an email from me last night with some more details about these instruments. The last portion of the show today, we can timestamp that so you can review the different software that we used. And if you'd like us to walk through step by step, let us know. For students, if you're still watching, still engaged, this is a warm-up for you to get a good sense of what we're going to do next Friday. So on the 22nd, it's going to be a bigger balloon. It's going to go farther. It's going to go higher. And then we're going to go back to this small balloon on the 29th. And on June.
There's a really lively conversation happening on YouTube.
What are they saying, Tony?
Should we send turtle in space?
Okay. A Ninja Turtle? What kind of turtle are we talking about? Well, what temperature is up there? Right? Is it warm or is it cold? Will something be alive? If you have a lower atmospheric pressure, does that mean it's harder to breathe? Or is it easier to breathe? Something you might want to think about. And so if you're going to send a living thing up there, what's the likelihood that it will survive? Right? Now, if you find that it will survive, then maybe we can send it up. But if you find that it can't, maybe we shouldn't, unless, now I would have to take that into consideration. If you found out that it wouldn't survive, if you could build a habitat for it to survive, and you could show us that it could work, then maybe we could do something like that.
You probably can't see this, Ted. I think they wouldn't. The conversation was potentially about it going, I don't know. I was thinking it was onto the space station, but maybe I did misunderstand, because they're talking about going into space.
Oh, okay.
And obviously, we're with the balloon launch, we're not going into space. We're just going into the same sort of altitude as planes.
So to put that in context, this balloon here is going to go up a few miles into the atmosphere, 4 or 5 miles, I guess. In order to get to space, there's an arbitrary line there where the Earth's atmosphere ends and the vacuum of space begins. But there's something called the Kármán line, which is about 62 miles from mean sea level into the atmosphere. If you get around 62 miles, I think almost everyone says you're technically in space. So we'd have to travel substantially farther. Now, our balloon is running on a lighter than atmospheric gas. It's running on a helium to lift. And if there is no more atmosphere around it, is it still going to lift? Is another question you might want to ask yourself, right? And if we use hydrogen, that's even a lighter gas. Our sun is fueled by hydrogen. That'll lift us higher. Will we still be able to leave? It could be that it's possible. It could be that it's not. Now, I kind of know the answer already, but I don't want to necessarily spill the beans. Ha ha, Leguminaut, little plug there. But it's a good investigation, good questions to ask. But how can you get to space in your lifetimes? Right now, as we're talking about it, something you've probably heard about Virgin Airlines. And Virgin, well, Sir Richard Branson also has like Virgin Records, but he has a company called Virgin Galactic. And if you're willing to pay a seat price of only $250,000, he's going to take you to space. And it's a space plane's going to go up. It won't go as high as 62 miles, but they're calling it leaving Earth. And you're going to be suborbital, and you're going to look down, not too dissimilar to the views we've gotten on some of our big balloons with the camera. Where you see a beautiful curve of the Earth and the clouds far below, and far below the clouds, so you might see villages or towns or cities or roads or forests and lakes. It's just really beautiful. And you start realizing we have just one world, this one planet. And it's going to be kind of cool to live on other planets, but there's so much we can do here to make this a better planet to live on. And we need your help. There's a lot of things we need to work on. And there's some fun stuff and there's some challenging things. So hopefully you can find something you get excited about. And there's a lot of teachers that would love to help you along that path. So I think we're pretty much half past our hour here. We're past our go time. So I think we'll sign off for now. You've got the link to track our balloon. I'd love to hear from you guys through your teachers. So send them a note where you think it's going to end today. When's the last transmission it will send? You saw that prediction. We can forward that prediction to you as well so you can look at it. But I'd love you to estimate where you think it might end its transmission today. It's relying on solar power, so when it can't get power from the sun anymore, it'll shut down. And here's a little hint, that won't be sunset. The power drops sooner than sunset because the sun is very low in the sky and it's not able to charge up our solar arrays. So it'll probably be turning off around 5-ish. It's just a guess. So if you want to try to estimate looking at that calculator where you think 5 o'clock would be and where you think it might end up being today, we'll find out. And the next question for you is, where do you think it's going to wake up on Saturday morning? Now, we've had some balloons that don't even wake up the next day. They were pretty much done for one day. But if it wakes up, where will it wake up? And where will it go tomorrow, Saturday? And where will it end the day? And do the same thing for Sunday. And when we talk again on Monday or Tuesday, it'll be really interesting to see where it is. Is it going to be slow down near Mexico? Is it going to be really fast up near Canada? At least that's what our model showed. I don't know. Let's find out. So anyway, you guys have a wonderful weekend. Thank you for joining us with Magnitude Live. If you're just thinking about the Leguminaut challenge, plenty of time to join us. Just need to plant something or do a little research. Help us know what's the right bean to send to space, or I should say legume, because there's more than just beans in that category. And join us next week for Magnitude 13. I believe we're going to have Frank Bauer from Eris join us, talk a little bit about ham radio, and he basically give us a nice intro to our around the world attempt #1. We'll see if we can't go around the world with that one. Frank is a very, very super knowledgeable guy. He works with talking to astronauts using ham radio, and he's like on the boards of all these important things. He also works with a group called AMSAT, Amateur Satellites, in which the AMSATs around the world, they build satellites that amateur radio operators can communicate with, which is super cool. I haven't done that yet. I think, Liam, you've been tracking some satellites, right?
Yes. Yeah. With even the International Space Station.
Yep.
So whenever the space station passes by, if you just have any ham radio, even a $30 ham radio from Amazon, this is a little bit more expensive than that, you can hear the space station beacon on a particular channel. You don't need you don't need any special equipment other than and pretty much you'll be able to hear it on a regular antenna on a simple ham radio thing under $30 from Amazon.
But that's not a regular antenna you got there.
Say that no, and you don't need to be ham radio certified in order to listen. So you've just got to, you're not allowed to press the transmit button. So if you have less than $30 to spare, you can find a ham radio and you just tune it to the right frequency and you can hear the space station as it goes by. It's the coolest thing.
That's one thing.
You have one of these, you can transmit.
Yeah. Well, let's end that here. But Liam, you and I should chat about pedagogically what we can do to introduce radio comms in our programming for the fall. I think that'd be a lot of fun.
Yeah, that would be cool.
Guys, have a wonderful weekend. Stay safe
Thank you, everybody. This is Ms. Hawker and Ms. Tate signing off. We just want to say thank you. It was great.
Catch up next
Thank
week.
you so much.
Thank you for coming.
Bye, thank you guys.
Take care, everyone.
Bye,
Bye
guys.
now.
Thank you.